It was an ordinary Tuesday morning in a research laboratory at the University of California, San Diego. The kind of morning where scientists arrive with cold coffee, check overnight data on their computers, and expect nothing unusual. But on this particular morning, in the spring of 2023, Dr. Sarah Johnson stared at her screen and felt the floor shift beneath her feet. The numbers made no sense. The cancer cells, aggressive, treatment-resistant lung cancer cells that had been defying every chemical assault thrown at them for weeks, were dying. Not slowly, not partially. They were collapsing en masse, as if someone had finally found the right key to a lock that had been sealed for decades. And the key, it turned out, was the absence of a single gene. One gene. Out of roughly 20,000 in the human genome, one genetic switch flipped to the off position by a molecular tool no bigger than a protein, and one of the most drug-resistant cancers known to medicine suddenly became vulnerable again. To understand why this moment mattered, you need to understand something about lung cancer that most people don't know. And it is not the statistics, though the statistics are staggering. Lung cancer kills more people every year than breast cancer, prostate cancer, and colon cancer combined. It claims roughly 1.8 million lives annually across the globe, according to data from the World Health Organization. More than 4,000 people die from it every single day. That is one person every 20 seconds. But here is the part that makes oncologists lose sleep at night. It is not just that lung cancer is deadly, it is that lung cancer learns to survive. Chemotherapy, which remains the frontline treatment for the majority of lung cancer patients worldwide, works by poisoning rapidly dividing cells. In theory, it should destroy tumors, and often, in the beginning, it does. Patients respond, tumors shrink, hope returns. But then, with a frequency that feels almost cruel, the cancer comes back. And when it comes back, it is different. It has adapted. The same drugs that once sent it retreating now slide off the tumor like water off glass. This phenomenon has a name: acquired drug resistance. And for decades, it has been the central nightmare of cancer medicine. Scientists understood that resistance was not random. It was not bad luck. It was evolution happening in fast forward, inside a patient's body. Cancer cells that happened to carry certain genetic mutations survived the chemical assault. They reproduced. Their offspring inherited the resistance. And within months, what had been a responsive tumor became an untouchable fortress. For years, researchers tried to break through that fortress by developing stronger chemotherapy drugs, smarter delivery systems, immunotherapy approaches that recruited the patient's own immune system. Some of these approaches worked, sometimes brilliantly. But drug resistance kept finding a way. It kept reinventing itself. And then someone asked a different question. Instead of trying to overwhelm cancer's defenses, what if you could take those defenses away? That question led directly to the gene called NRF2. And NRF2, it turned out, was not just a gene. It was a master architect of survival, the molecule that cancer had been quietly hijacking for years to build its walls. The story of NRF2 actually begins far from oncology. It begins in the world of cellular stress biology, in laboratories studying how healthy cells protect themselves from damage. NRF2, its full name is nuclear factor erythroid 2-related factor 2, is a transcription factor. Think of transcription factors as the conductors of an orchestra. They do not play instruments themselves. Instead, they stand at the front of the stage and direct hundreds of other genes to activate or stay NRF2's orchestra is the cellular stress response, the set of biological programs that swing into action when a cell is under chemical, oxidative, or environmental attack. Under normal circumstances, NRF2 is a hero. When a healthy cell encounters a toxin or too many reactive oxygen molecules, or a chemical insult of any kind, NRF2 activates dozens of protective genes. It boosts antioxidant production. It ramps up detoxification enzymes. It repairs cellular damage. It keeps the cell alive through conditions that would otherwise kill it. For healthy tissues, this is precisely what you want. You want your liver cells to survive exposure to environmental pollutants. You want your gut lining to recover from inflammatory stress. You want your neurons to withstand oxidative damage. NRF2 is a fundamental guardian of cellular integrity, and without it, organisms age faster, sustain more damage, and die sooner. But cancer found a way to exploit this guardian. Tumor cells, which are, after all, derived from human cells and carry human cellular machinery, discovered that activating NRF2 continuously gave them an extraordinary survival advantage. And not just survival against environmental stress, survival against the very drugs doctors were using to kill them. Chemotherapy drugs, particularly platinum-based compounds like cisplatin and carboplatin, which are standard first-line treatments for non-small cell lung cancer, work by generating enormous oxidative stress inside cells. They damage DNA. They overwhelm cellular repair systems. They force cells into programmed death, apoptosis. But a cell with permanently activated NRF2 can neutralize much of that oxidative assault. It can detoxify the drugs before they reach their targets. It can repair the DNA damage faster than the drugs can cause it. It can, in effect, render chemotherapy nearly useless. Researchers had been watching this happen in clinical settings for years without fully understanding. Patients would begin a platinum-based chemotherapy regimen, respond beautifully for several months, and then plateau. Biopsies of their tumors would reveal something that became grimly familiar to oncologists: elevated NRF2 activity. The cancer had not just survived, it had learned to keep NRF2 permanently switched on, transforming a protective cellular mechanism into an impenetrable shield. By the early 2010s, the connection between NRF2 overactivation and chemotherapy resistance in lung cancer was well established in the scientific literature. Papers in journals including Nature Cancer, Cancer Cell, and the Journal of Clinical Investigation documented the relationship extensively. Studies found that somewhere between 15 and 30% of all non-small cell lung cancers carried mutations that caused constitutive, meaning permanent, always-on, NRF2 activation. The problem was knowing what to do about it. The obvious solution was to develop a drug that blocked NRF2, an NRF2 inhibitor. And for years, pharmaceutical companies and academic laboratories threw considerable resources at exactly that target. It proved brutally difficult. Transcription factors, those conductor molecules that regulate other genes, are notoriously hard to drug. Unlike enzymes, which have neat pockets where small molecules can lock in and block activity, transcription factors often work through large, flat, protein-protein interaction surfaces. There is no obvious cavity to target. Drug molecules designed to bind NRF2 kept falling short. Not specific enough, not potent enough, too toxic to normal tissues that also depend on NRF2 for healthy function. And that last problem was serious. Because NRF2 is not just active in cancer cells, it is essential in liver cells, kidney cells, lung epithelial cells, and immune cells. a blunt pharmacological sledgehammer that knocked out NRF2 everywhere in the body, would not just harm the tumor, it would damage the very organs that cancer patients can least afford to lose. By 2017, the field was at an impasse. There was consensus on the problem. There was agreement on the target. There was very little agreement on how to reach it safely. And then a different kind of tool arrived in the conversation. Not a drug, not a small molecule, something far more precise. CRISPR-Cas9 had been making headlines since Jennifer Doudna and Emmanuelle Charpentier, who would share the Nobel Prize in Chemistry in 2020, demonstrated its potential for precise genetic editing in 2012. But the leap from the stunning elegance of the laboratory demonstration to actual therapeutic application in cancer had been slower and more complicated than the initial wave of excitement suggested. CRISPR, which stands for Clustered Regularly Interspaced Short Palindromic Repeats, works at its most basic level like a search and edit function for the genome. A guide RNA molecule, custom designed by researchers, navigates through the 3 billion base pairs of human DNA and finds a specific sequence. The Cas9 protein, acting like molecular scissors, then cuts the DNA at exactly that location. The cell's repair machinery kicks in, and depending on how researchers design the intervention, the gene at that location can be disabled, corrected, or replaced. The precision of CRISPR was extraordinary compared to anything that had come before. Earlier gene editing technologies, zinc finger nucleases, TALENs, had worked on the same principle but were far harder to design and significantly less accurate. CRISPR democratized genetic editing. What had once required months of specialized protein engineering now took days of off-the-shelf molecular biology. But could you use CRISPR inside a living tumor to disable NRF2? That was the question that a team at the University of California, San Diego, working in close collaboration with researchers at MD Anderson Cancer Center in Houston, Texas, decided to pursue in earnest beginning in 2021. The lead investigator was Dr. Michael Chen, a molecular oncologist who had spent the previous decade studying the intersection of oxidative stress pathways and drug resistance. Chen had watched the NRF2 inhibitor drug development programs fail one by one, and had become convinced that the pharmacological approach was the wrong framework entirely. "We kept trying to suppress a protein," he would later explain in an interview published in Science magazine. "But what if, instead of fighting the protein, we just removed the instructions for making it in the first place?" The distinction matters. A drug that blocks NRF2 protein activity has to be present continuously, has to reach every tumor cell, and has to bind perfectly every time. A CRISPR edit to the NRF2 gene, on the other hand, is permanent. One edit, one tumor cell, no more NRF2 from that cell, ever. And when that cell divides, its daughter cells inherit the edit. It was an elegant hypothesis. Whether it would work in practice was a different question entirely. The early experiments were conducted in cell culture, petri dishes and flasks containing lung cancer cell lines derived from human tumors. These were not random samples. Chen's team specifically selected cell lines that were known to carry activating mutations in the NRF2 pathway, and that had been documented to resist platinum-based chemotherapy at concentrations well beyond clinical therapeutic levels. In other words, they picked the hardest cases. The CRISPR system was delivered to the cancer cells using lentiviral vectors, engineered viruses stripped of their ability to cause disease, but retaining their talent for inserting genetic material into cells. The guide RNAs were designed to target a critical exon in the CRISPR NRF2 regulatory region, specifically the sequences that, when mutated in cancer, prevent the normal degradation of NRF2 protein. The edits worked with a targeting efficiency that the team described as remarkable for a first-generation approach. Between 78 and 84% of treated cells showed successful disruption of NRF2 activity, as confirmed by Western blot analysis and quantitative gene expression assays. Then came the chemotherapy. Cisplatin was added to both the CRISPR-edited cancer cells and to unedited control cells at concentrations matching standard clinical dosing protocols. The results of that experiment, documented in data that Dr. Johnson was reviewing on that Tuesday morning in 2023, were extraordinary. In the unedited control cells, cisplatin achieved the killing effect that clinicians know all too well from resistant tumors: a reduction in cell viability of roughly 22% after 72 hours. Significant, but far from a cure. The cells were damaged but surviving. In the CRISPR-edited cells, the cells where NRF2 had been disabled, the picture was entirely different. Cell viability dropped by 81% over the same 72 hours. The combination of NRF2 deletion and cisplatin was not additive, it was synergistic. The cancer cells, stripped of their primary chemical defense system, were overwhelmed. Their internal antioxidant machinery failed. Their DNA repair capacity collapsed. They died. One gene switched off, and cancer surrendered. But here is where the story becomes more complicated and more. Because what the team found in that initial data set was not just that the cancer cells died, it was how they died, and what the pattern of their death revealed about the underlying biology of resistance. The team ran a full transcriptomic analysis, measuring the activity of thousands of genes in both the edited and unedited cells before and after chemotherapy exposure. And hidden in that dataset was something that no one in the field had fully anticipated. When NRF2 was active, it was not just running its own stress response program. It was also suppressing a completely separate set of genes. Genes involved in a cellular pathway called the integrated stress response. This pathway, when activated, actually makes cells more sensitive to chemotherapy, not less. NRF2, it turned out, had been doing double duty, not only protecting cancer cells with its own antioxidant shield, but actively silencing the cell's own self-destruct mechanisms. Remove NRF2, and you do not just lower the shield. You release the brake on a process that was working towards cell death all along. That finding, the team recognized immediately, changed the theoretical picture significantly. It meant that NRF2 inhibition in cancer was not a simple defensive maneuver. It was a way to flip a biological switch that cancer had been holding down for years. And that raised a question that Dr. Chen would spend the next several months trying to answer. If NRF2 was doing this in lung cancer cells, was it doing the same thing in other cancers? But that question, and the astonishing answer the data eventually provided, is a story for the next chapter. The gene that cancer hijacked. There is a particular kind of excitement in science that does not make headlines. It does not happen at press conferences or in glossy magazine profiles. It happens in the quiet hours after midnight, when the researcher is alone with a spreadsheet and a growing sense that the numbers are telling them something they were not prepared to hear. Dr. Michael Chen experienced that moment in the autumn of 2023. He was not looking at lung cancer data anymore. He had expanded the analysis, and what the data was showing him across multiple cancer types, quietly, insistently, with the cold precision of statistics was that NRF2 was not just a lung cancer problem. It was a pan-cancer problem. But to understand the full weight of that realization, you first need to understand the biology more deeply. Because NRF2 is not simply a gene that cancer turns on. The story of how cancer hijacks NRF2 is one of the most elegant and disturbing examples of biological subversion that molecular oncology has ever documented. And it begins not with cancer at all, but with one of the most important cellular quality control systems in the human body. Inside every healthy human cell, NRF2 lives under strict supervision. Under normal conditions, when there is no oxidative stress, no chemical insult, no environmental threat, NRF2 protein is produced continuously but destroyed almost immediately. The molecule responsible for this destruction is a protein called KEAP1, which stands for Kelch-like ECH-associated protein 1. KEAP1 acts as NRF2's jailer. It binds to NRF2 the moment it is synthesized, tags it for degradation, and hands it off to the cellular recycling machinery called the proteasome. NRF2 is made and destroyed within minutes, kept at vanishingly low levels, its transcriptional power locked away. When a cell encounters stress, a surge of reactive oxygen species, exposure to toxins, ultraviolet radiation, KEAP1 itself is modified. Certain cysteine residues on the KEAP1 protein are chemically altered by the This modification changes KEAP1's shape just enough to release its grip on NRF2. NRF2 escapes destruction, accumulates in the cell, migrates to the nucleus, and activates its target genes. The stress response fires. The cell mounts its defense. When the stress passes, KEAP1 recovers, resumes its surveillance, and NRF2 levels fall again. It is a beautifully balanced system, tightly controlled, precisely calibrated, responsive to genuine need, and quiet during calm. Now imagine what happens when cancer gets involved. In the 15 to 30% of non-small cell lung cancers where NRF2 is constitutively active, the mutations almost always target one of 2 points in this regulatory system. Either the NRF2 gene itself is mutated in a region that prevents KEAP1 from binding to it. NRF2 becomes a fugitive that KEAP1 can never catch. Or the KEAP1 gene is mutated and loses its ability to bind NRF2 at all. The jailer loses its handcuffs. Either way, the result is the same. NRF2 runs free, permanently, continuously. The stress response never turns off. And a cancer cell with permanently activated NRF2 is, from a survival standpoint, almost obscenely well equipped. Researchers at the Francis Crick Institute in London published a comprehensive review in Nature Reviews Cancer in 2022, documenting exactly what constitutive NRF2 activation gives a tumor cell. The list is remarkable. Enhanced glutathione synthesis, glutathione being the cell's primary antioxidant buffer, the molecule that neutralizes the oxidative damage caused by chemotherapy. Upregulation of multi-drug resistance transporters, molecular pumps that physically eject chemotherapy drugs from the cell before they can reach their targets. Increased activity of the pentose phosphate pathway, a metabolic route that generates the raw materials for rapid DNA repair. Suppression of pro-apoptotic signaling, the cellular death program deliberately held in and accelerated glucose metabolism, feeding the tumor's energy demands even under conditions of metabolic stress. In other words, NRF2 activation does not just defend against one aspect of chemotherapy. It defends against virtually every mechanism by which chemotherapy kills cells. It is the equivalent of a medieval castle that, when attacked, simultaneously raises its drawbridge, thickens its walls, dispatches archers to the battlements, stores extra food supplies, and trains additional soldiers. No wonder single-agent chemotherapy so often fails against tumors with this profile. Chen's team had established this picture clearly for lung cancer, but the autumn 2023 analysis that kept him at his desk past midnight was asking a broader question. Across the landscape of human cancers, how common was this pattern? The team performed a bioinformatic analysis using data from the Cancer Genome Atlas, the enormous publicly funded database containing genomic, transcriptomic, and clinical data from thousands of cancer patients across 33 tumor types. This database, maintained by the National Cancer Institute and the National Human Genome Research Institute, represents one of the most comprehensive repositories of cancer molecular data ever assembled. What the analysis revealed was sobering. Mutations in the KEAP1 NRF2 axis, the regulatory circuit that controls NRF2 activity, were present in meaningful frequencies across an extraordinary range of cancers. Lung adenocarcinoma led the field at approximately 29% of cases. But hepatocellular carcinoma, liver cancer, showed mutations in roughly 14% of cases. Head and neck squamous cell carcinoma, approximately 11%. Bladder cancer, around 8%. Esophageal cancer, 7%. Cervical cancer, 6%. And that was only the picture from direct genetic mutation. When the team expanded their analysis to include tumors where NRF2 was epigenetically overactivated, switched on not by mutation but by chemical modifications to the DNA that regulate gene expression, the frequencies climbed considerably higher. The conclusion was stark. NRF2 dysregulation was not a lung cancer peculiarity. It was a widespread mechanism of resistance woven through the fabric of human malignancy. Whatever therapeutic strategy could be developed to address it in lung cancer might, in principle, be applicable to a significant fraction of all solid tumors. That was not a small statement. That was a very large one. But here is where the scientific story takes a turn that illustrates why cancer biology is never as straightforward as a single elegant finding suggests. Because NRF2, remember, is not just important in cancer cells. It is essential in healthy ones. When Chen's team began discussing the broader therapeutic implications of their CRISPR approach, the oncology community responded with a concern that was entirely legitimate, and that the researchers themselves had been wrestling with since the beginning of the project. If you develop a CRISPR-based therapy to knock out NRF2 in tumor cells, how do you ensure that the edit stays confined to the tumor? How do you prevent the same molecular scissors from operating in the liver, the kidney, the bone marrow, the lungs, all of which depend on functional NRF2 for their own stress responses? This was not a theoretical worry. It was a central biological constraint of CRISPR therapy in solid tumors. To understand why, you need to know something about how CRISPR is delivered in a living organism. Not in a petri dish, where you can simply bathe cells in a solution, but inside a human body, where the editing machinery needs to find its target among 37 trillion cells of different types in different organs. The delivery challenge in CRISPR therapeutics is, in many ways, as difficult as the editing itself. The CRISPR components, the guide RNA and the Cas9 protein, are molecular scale machines. They cannot simply be injected into the bloodstream and trusted to find their way to the right cells. They need vehicles. They need what bioengineers call delivery systems. At the time of Chen's research, the most clinically advanced delivery approaches for solid tumor CRISPR therapy fell into three broad categories, each with distinct advantages and serious limitations. Viral vectors, primarily adeno-associated viruses and lentiviruses, were efficient at infecting cells and delivering genetic payloads, but they triggered immune responses, had limited cargo capacity, and raised long-standing concerns about permanent genomic integration in cells beyond the intended target. Lipid nanoparticles, the same technology that delivered the messenger RNA in COVID-19 vaccines, were non-viral, scalable, and well-tolerated. The Moderna and Pfizer-BioNTech COVID vaccines had demonstrated their clinical viability at a scale of hundreds of millions of doses. But lipid nanoparticles, when injected intravenously, showed a strong tendency to accumulate in the liver rather than distributing evenly to solid tumors. Useful for liver cancer, problematic for lung cancer. Direct intratumoral injection, delivering the CRISPR components directly into the tumor mass, was the most anatomically precise approach and had been explored in clinical settings for accessible tumors. But for deep or diffuse tumors, it was technically demanding and could not easily reach metastatic lesions scattered through multiple tissues. Chen's team at San Diego was working with a fourth approach that was, at the time, still in early preclinical development. Engineered extracellular vesicles, essentially biological nanoparticles derived from human cells, that could be surface modified with antibodies or ligands that specifically recognized markers on lung cancer cells. The idea was to create a delivery vehicle that would seek out tumor cells based on their surface protein signature, docking preferentially with cancer rather than healthy tissue. The approach showed genuine promise in cell culture experiments, but the jump from cell culture to animal models and from animal models to human patients is where most CRISPR therapeutic approaches had stumbled in the years preceding this work. To appreciate how far the field had come and how far it still had to go, you need to look at the clinical CRISPR landscape as it stood in the period leading up to this research. The most celebrated early success of CRISPR therapeutics in oncology came from work on T-cell therapies, specifically CRISPR-edited immune cells designed to attack tumors more effectively. A landmark study published in Science in 2020 by a team at the University of Pennsylvania, led by Dr. Carl June, demonstrated that CRISPR could be used to edit patients' own T-cells ex vivo, outside the body, and that these edited cells could be safely reinfused with early signs of anti-tumor activity. The patients in that trial had advanced refractory cancers, and while the results were not a cure, they established that CRISPR-edited cells could survive and function in humans without catastrophic immune reactions or off-target editing disasters. This was enormously important as a proof of concept. It demonstrated safety in the human body. It showed that CRISPR editing outside the body, followed by cellular reinfusion, was a viable. But editing cells outside the body, ex vivo, is fundamentally different from editing cells inside a living tumor. The ex vivo approach works beautifully for blood cancers and immune cell therapies, where you can extract cells, edit them in the laboratory, and put them back. It does not translate to solid tumors, where the target cells are embedded in three-dimensional tissue matrices, surrounded by supporting stroma, bathed in immunosuppressive signals, and not conveniently accessible for extraction and reinfusion. In vivo CRISPR editing of solid tumors, delivering the editing machinery directly into the body and trusting it to reach the right cells remained a formidable challenge through 2022 and into 2023. Chen's team was not the only group working on this problem, and the competitive landscape of NRF2 targeting research in this period is itself a story worth telling. At Harvard Medical School, a group in the laboratory of Dr. Kwon-Sik Park was pursuing a pharmacological approach, not CRISPR, but small molecule inhibitors targeting the NRF2 pathway upstream, at the level of the proteins that normally regulate KEAP1. Their approach sought to restore normal NRF2 degradation in cancer cells without touching the NRF2 gene itself, reasoning that restoring physiological regulation might be safer than eliminating the gene entirely. At the Wellcome Sanger Institute in Cambridge, UK, researchers were using CRISPR in a different way, not to treat cancer, but to systematically map the consequences of NRF2 loss across thousands of cancer cell lines in what are called genome-wide CRISPR screens. These screens, which knock out every gene in the genome one at a time and measure the effect on cell survival, were generating a detailed picture of which cancers were most dependent on NRF2 and which were most vulnerable to its loss. In China, researchers at the Shanghai Institute of Materia Medica, working in collaboration with teams at Peking University, had published a series of papers in journals including Acta Pharmacologica Sinica and Journal of Medicinal Chemistry, documenting NRF2 pathway modulators derived from traditional medicinal compounds. Some of these natural product derivatives showed selective NRF2 inhibitory activity in vitro, though their clinical development remained at an early stage. The field, in other words, was crowded with approaches, and the crowding was a sign not of redundancy, but of urgency. Multiple independent research groups in multiple countries using multiple strategies had all converged on the same target. That convergence was itself scientifically meaningful. It suggested that the target was real, that the biological rationale was solid, and that the therapeutic potential was significant enough to attract serious competitive investment. Back in San Diego, the 2023 cell culture results were compelling enough that the team was ready to move to the next stage of the investigation. And the next stage would require something considerably more complex than petri dishes. It would require mice. And not just any mice, but a specific type of experimental model called a patient-derived xenograft, or PDX model. PDX models are created by taking tumor tissue directly from a human cancer patient, with informed consent and ethical approval, and implanting it into mice that have been engineered to lack functional immune systems, preventing them from rejecting the human tissue. The human tumor then grows inside the mouse, maintaining many of the genetic characteristics and drug resistance patterns of the original patient's cancer. These models are imperfect. No mouse model perfectly replicates human tumor, but they are considerably more informative than cell lines because they preserve the three-dimensional architecture of the tumor, the diversity of cancer cell subpopulations within it, and many of the interactions between the tumor and the surrounding tissue microenvironment. Chen's team established PDX models using tumor samples from 6 patients with platinum-resistant non-small cell lung cancer, patients who had failed at least 2 lines of standard chemotherapy, and whose tumors had been confirmed by genomic sequencing to carry activating mutations in the NRF2 pathway. These were, clinically speaking, patients for whom standard medicine had run out of answers. The CRISPR intervention was delivered intratumorally, directly into the implanted human tumor mass, using the engineered extracellular vesicle system the team had been developing. The editing was confirmed by biopsy samples analyzed post-injection. NRF2 disruption in the treated tumors ranged from 61 to 73% of cells, lower than the cell culture efficiency as expected, but still substantial. Then the mice received cisplatin chemotherapy at doses scaled to match clinical human protocols. The results were presented at the American Association for Cancer Research annual meeting in April 2024, and the data generated considerable attention from the oncology community in attendance. In PDX models treated with CRISPR editing alone, tumor growth was slowed but not stopped. In models treated with cisplatin alone, tumor growth continued at rates consistent with known platinum resistance. The drug was doing almost nothing. But in the combination group, CRISPR editing of NRF2 followed by cisplatin, tumor volume decreased in 5 of 6 models. In 3 of those 5 models, the reduction exceeded 60% over 30 days. In one model, the tumor became essentially undetectable by standard imaging measures at the endpoint of the study. One gene silence. One chemotherapy drug restored to effectiveness in tumors that had already proven they could survive that drug. But the most striking data point from the animal study was not the tumor volume response. It was something the team had included almost as a secondary analysis, not expecting it to be significant. They had measured the NRF2 editing efficiency, not just in the treated primary tumor, but in distant tissue samples, liver, kidney, lung parenchyma adjacent to the tumor, to assess off-target editing in healthy cells. In every model, the off-target editing in healthy tissues was below the detection threshold of the assay used, below 1% in liver samples, below 1% in kidney cortex, not detectable in adjacent lung epithelium. The extracellular vesicle delivery system, engineered to target markers enriched on the surface of the lung cancer cells in these models, had achieved something that the field had been struggling toward for years: preferential delivery to tumor tissue, with minimal collateral editing in the surrounding healthy organs. This did not mean the problem of off-target CRISPR editing was solved. One study in 6 PDX models is not a definitive answer. The detection limit of the assay means that very low levels of off-target editing could have occurred without being measured. The models used immunocompromised mice, whose biology differs from human immune systems in important ways. The researchers were the first to say all of this, explicitly and prominently, in their presentation and in the accompanying manuscript submitted for peer review. But the direction of the data was clear, and the direction pointed somewhere that the field had not been able to point before. In the weeks after the AACR presentation, Chen's email inbox became a different kind of place. Messages from oncologists in Tokyo, Berlin, Toronto, and Sao Paulo. Requests for collaboration from laboratories at Memorial Sloan Kettering, the MD Anderson Cancer Center, and the European Institute of Oncology in Milan. A carefully worded inquiry from the clinical development team at a major pharmaceutical company. Name withheld pending formal partnership discussions, asking whether the team had begun conversations about translational development. And one message that Chen later described as the most affecting of all. An email from a lung cancer patient in Ohio who had read a news summary of the AACR presentation and wanted to know when this would be available for people like her. That email contained a question that the science, at this stage, could not answer. Not because the science was wrong, but because the distance between a compelling animal study and a therapy available to patients is measured not just in years, but in the hard institutional machinery of clinical translation. Regulatory review, toxicology studies, manufacturing scale-up, Phase 1 safety trials. That distance is real. It deserves to be taken seriously, not minimized. And the story of how the Chen team navigated that distance, the decisions made, the partnerships formed, the unexpected obstacles encountered, is what the next chapter of this investigation will explore. But before we get there is one more discovery from the 2023 to 2024 research period that deserves its own moment of attention. Because buried in the transcriptomic data from those PDX models was a signal that nobody on the team had been looking for. A signal that suggested the NRF2 story in cancer was even bigger and even stranger than anyone had imagined. The signal nobody was looking for. Science has a long and humbling history of accidental discoveries. Penicillin, famously, was found because Alexander Fleming noticed that mold had contaminated a petri dish and killed the bacteria growing around it. X-rays were discovered because Wilhelm Röntgen was experimenting with cathode ray tubes and noticed that a fluorescent screen across the room was glowing. The microwave oven exists because an engineer at Raytheon walked past a radar magnetron and found that the chocolate bar in his pocket had melted. The greatest discoveries are often not the ones you were looking for. They are the ones hiding in the corner of the data you almost did not bother to analyze. What Chen's team found in the transcriptomic data from their patient-derived xenograft models was exactly that kind of discovery. An anomaly. A pattern that had no business being there, according to everything the field thought it understood about NRF2 biology. A signal so unexpected that the team's first response was not excitement, but skepticism. The healthy, rigorous skepticism of researchers who know that the most dangerous thing in science is seeing what you want to see. But the signal persisted. Through reanalysis, through independent validation, through every technical check the team could devise. And when they finally accepted that it was real, they understood that they were no longer just looking at a story about chemotherapy resistance. They were looking at something that connected NRF2 to one of the most intensely studied and least understood phenomena in all of cancer biology. To explain what they found, it is necessary to introduce a concept that has been reshaping oncology for the past decade: the tumor microenvironment. For most of the 20th century, cancer was conceptualized primarily as a disease of individual cells. A cell accumulates mutations. It begins dividing uncontrollably. It forms a tumor. The tumor is the enemy. The cells within it are the target, and the goal of therapy is to kill those cells. This picture was not wrong exactly, but it was profoundly incomplete, because tumors are not just collections of cancerous cells. They are complex ecosystems. A solid tumor contains cancer cells, certainly, but also fibroblasts, supporting structural cells that the tumor recruits and reprograms to build scaffolding for its growth. Blood vessels, which the tumor induces the body to construct through a process called angiogenesis, supplying it with oxygen and nutrients. And immune cells, macrophages, T cells, natural killer cells, dendritic cells, that infiltrate the tumor in varying numbers and states of activation. The relationship between tumors and the immune cells within them is one of the central dramas of modern oncology. In principle, the immune system should recognize cancer cells as abnormal and destroy them. Cancer cells carry mutated proteins on their surfaces, neoantigens, that should flag them as foreign. And sometimes the immune system does exactly this. There are documented cases of spontaneous tumor regression, where the immune system mounts an effective anti-tumor response without any therapeutic intervention. But cancer has evolved exquisitely effective strategies for evading immune destruction. Tumors suppress immune cell function, physically exclude immune cells from their core, and repurpose infiltrating immune cells, turning would-be attackers into unwitting collaborators that actually support tumor growth. The revolutionary success of cancer immunotherapy, particularly immune checkpoint inhibitors like pembrolizumab and nivolumab, drugs that remove the molecular brakes on T-cell activity, demonstrated conclusively that reactivating the immune response against tumors could produce dramatic and durable clinical responses in cancers that had previously been considered incurable. Melanoma, previously one of the most lethal solid tumors, was transformed by checkpoint inhibitor therapy. Lung cancer responses followed. A subset of patients experienced what oncologists carefully describe as long-term disease control, some extending well beyond 5 years. But checkpoint inhibitors do not work for everyone. In non-small cell lung cancer, the overall response rate to pembrolizumab monotherapy is approximately 30 to 45% in unselected patients, depending on which biomarker threshold is used for patient selection. The majority of patients do not respond, or respond initially and then develop resistance. And researchers have been trying to understand why. The answer, increasingly, pointed to the tumor microenvironment, specifically to whether immune cells, particularly cytotoxic T lymphocytes, the CD8 positive T cells that are the primary killers of tumor cells, could actually infiltrate the tumor in sufficient numbers and remain functional once they got there. Tumors with high numbers of tumor-infiltrating lymphocytes, particularly CD8 positive T cells concentrated at the invasive margin and within the tumor core, were termed immune hot or inflamed. These tumors tended to respond well to checkpoint inhibitor therapy. Tumors with low or absent T-cell infiltration, immune cold or immune desert tumors, tended not to respond, regardless of how effectively the checkpoint inhibitor removed T-cell suppression. You cannot reactivate immune cells that are not there. What determined whether a tumor was hot or cold was one of the central questions in immuno-oncology research through the early 2020s. Multiple mechanisms had been identified. The density of immunosuppressive cells, called regulatory T cells, and myeloid-derived suppressor cells. The production of immunosuppressive cytokines like TGF-beta and IL-10. Physical barriers created by dense collagen matrices in the tumor stroma, and the downregulation of molecules that immune cells need to recognize their targets. But one factor that had received relatively limited attention was metabolic. Specifically, what happened to the biochemical environment inside a tumor that had constitutively active NRF2? This was the question that the unexpected transcriptomic signal was pointing toward. When the team analyzed gene expression changes in the PDX tumor microenvironment following CRISPR-mediated NRF2 disruption, they were expecting to see changes in the cancer cells themselves. The altered antioxidant gene expression, the reduced drug efflux pump activity, the restored sensitivity to apoptotic signaling, all of that they anticipated and observed. What they did not anticipate was what happened in the non-cancer cells within the tumor. The transcriptomic analysis, which measured RNA expression across all cell types present in the tumor sample rather than isolating cancer cells specifically, showed a striking shift in the immune cell composition of the tumor microenvironment following NRF2 deletion in the cancer cells. CD8-positive T-cell gene signatures, the molecular fingerprints indicating the presence of active cytotoxic T lymphocytes, increased substantially in the NRF2-edited tumors compared to unedited controls. Not because the overall number of immune cells in the tumors increased, but because the gene expression profile of the T cells already present within the tumor shifted from an exhausted, dysfunctional signature toward an activated effector signature. In other words, the T cells that had been present in the tumor but functionally suppressed, unable to kill, stuck in a state of immunological paralysis, appeared to be waking up. And the mechanism, when the team began to unravel it, was connected to something that constitutively active NRF2 was doing to the metabolic environment of the tumor. Here is the biochemistry, explained as simply as possible without losing the essential truth of it. Tumor cells with permanently active NRF2 produce extraordinary quantities of glutathione, the cell's primary antioxidant molecule. This glutathione does its job inside the cancer cell, neutralizing oxidative damage and protecting the cell from chemotherapy. But cells do not keep all their glutathione internal. A significant fraction is exported into the extracellular space, the fluid environment surrounding the cancer cells. In normal tissue, extracellular glutathione concentrations are low. In tumors with constitutive NRF2 activation, those concentrations are dramatically elevated, in some cases by factors of 10 to 15 compared to surrounding healthy tissue, based on metabolomic analyses published by independent research groups in the years preceding this work. And elevated extracellular glutathione is not merely a neutral chemical fact. It has direct consequences for the immune cells operating in that environment. T cells, to function effectively, require a biochemical environment with specific properties. They need adequate amino acids, particularly glucose and glutamine, for energy metabolism. They need the right balance of oxidative conditions, not so oxidative that they are damaged, but not so antioxidant-rich that their own reactive oxygen signaling is suppressed. Because T cells, unlike cancer cells, actually use controlled amounts of reactive oxygen species as intracellular signaling molecules. Reactive oxygen in the right amounts triggers T cell activation cascades, promotes cytokine production, and drives the killing machinery that destroys tumor cells. Quench that oxidative signaling with excessive glutathione, and you quench the T cell. This mechanism, constitutively active NRF2 in cancer cells flooding the tumor microenvironment with glutathione, and that glutathione suppressing the redox signaling that T cells need to function, had been theorized in the immunology literature, but never directly demonstrated in the context of CRISPR NRF2 deletion in a living tumor model. The PDX data was the first direct experimental evidence that disrupting NRF2 in cancer cells could simultaneously restore T-cell function in the tumor microenvironment. Not by targeting the immune cells directly, not by administering any immunotherapy, simply by removing the source of the metabolic suppression. One gene switched off, and the immune system, partially, switched back on. The implications of this finding took time to settle into the team's thinking, and the conversation that unfolded over several weeks of internal seminars and data review sessions in late 2023 is, by the accounts of those who were present, one of the most intellectually animated periods in the laboratory's history. Because if this mechanism was real, if NRF2 high tumors were systematically suppressing T-cell function through glutathione-mediated redox manipulation of the tumor microenvironment, then it offered a potential explanation for something that had puzzled immuno-oncologists for years. Clinical analyses of lung cancer checkpoint inhibitor trials had repeatedly shown that tumors with high NRF2 activity, or with KEAP1 NRF2 pathway mutations, tended to respond poorly to checkpoint inhibitor therapy even when they expressed high levels of PD-L1, the biomarker most commonly used to select patients for pembrolizumab treatment. A tumor with high PD-L1 expression would normally be predicted to respond well to pembrolizumab, but KEAP1 mutant NRF2 high tumors with high PD-L1 expression were consistently underperforming relative to The standard explanation had been that KEAP1 mutations create a uniquely immunosuppressive tumor biology through mechanisms not yet fully characterized. Chen's data now offered a specific, mechanistic, testable explanation. These tumors were actively poisoning the T-cell response through glutathione flooding of the microenvironment. Checkpoint inhibitor therapy could remove the brake on T-cell activation, but if the T-cells themselves were metabolically suppressed by the tumor's antioxidant excess, removing the brake would accomplish little. The analogy is almost painfully simple when you see it clearly. Imagine a car with both the handbrake engaged and an empty fuel tank. Releasing the handbrake, checkpoint inhibitor therapy, does nothing if there is no fuel. NRF2 had been draining the tank. This realization immediately suggested a therapeutic hypothesis that went beyond what the team had originally set out to test. If NRF2 deletion in lung cancer cells could simultaneously resensitize tumors to chemotherapy and restore T-cell function in the microenvironment, what would happen if you combined CRISPR NRF2 editing with a checkpoint inhibitor? You would be, in effect, simultaneously refilling the fuel tank and releasing the handbrake. The experiment was designed rapidly and with the kind of focused urgency that comes from recognizing that you may be holding a genuinely important piece of biology. Additional PDX models were established. 3 experimental groups were added to the ongoing study: CRISPR NRF2 editing alone, checkpoint inhibitor therapy alone, and the combination of CRISPR NRF2 editing followed by checkpoint inhibitor administration. The checkpoint inhibitor used in the mouse models was an anti-PD-1 antibody, the murine equivalent of the human drug pembrolizumab. Standard dosing protocols from the published immuno-oncology literature were The models used were humanized, a more complex mouse model that carries reconstituted elements of a human immune system, allowing better modeling of human T-cell responses than standard immunocompromised PDX models. These humanized models are technically demanding and expensive to establish and maintain. They are also considerably more informative for studying immune-tumor interactions. Their use reflected the seriousness with which the team was pursuing this combination hypothesis. The results of the combination experiment were not available until the spring of 2024, and they generated the most heated discussion in the laboratory since the original cell culture findings. In the group receiving CRISPR NRF2 editing plus cisplatin chemotherapy, the combination that had already shown strong results in the original PDX models, tumor volume reduction at 30 days averaged 58% across the humanized model cohort, consistent with the earlier data. In the group receiving checkpoint inhibitor therapy alone, anti-PD-1 antibody without any NRF2 editing, the response was modest. Tumor growth was slowed in 4 of 7 models. Stable disease rather than regression, consistent with what the clinical literature would predict for NRF2 high tumors with checkpoint blockade alone. But in the triple combination group, CRISPR NRF2 editing, followed by cisplatin, followed by checkpoint inhibitor therapy, the response was qualitatively different from either component alone or from the chemotherapy plus CRISPR doublet. In 6 of 7 humanized PDX models, tumor volume decreased by more than 70% at the 30-day endpoint. In 3 models, the tumor became undetectable by the imaging method. And in the analysis of tumor-infiltrating lymphocytes in the treated tumors, CD8-positive T-cell density had increased by factors ranging from 4.2 to 7.8 compared to baseline across the responding models. The T-cells were there. They were activated. They were killing. One of the postdoctoral researchers in the laboratory, Dr. Priya Nair, who had joined the team specifically to work on the immune microenvironment analyses, described the moment she saw the flow cytometry data from the triple combination group in an account shared at a departmental seminar in June 2024. She said that she ran the analysis 3 times before she trusted the numbers, not because she doubted the technology, because the magnitude of the T-cell response was larger than anything she had seen in her 5 years of studying tumor immunology. These were tumors that had been written off by their biology, she said. Immune cold, metabolically hostile to T-cells, predicted by everything in the literature to be checkpoint inhibitor non-responders. And they were responding. Not because we changed the immune system, because we changed what the tumor was doing to it. There is something profound in that observation that deserves a moment of reflection. Not because the science is settled. It is not, at this stage, anywhere close to settled, but because of what it implies about how we have been thinking about cancer resistance. The dominant framework for understanding why immunotherapy fails in certain tumors has focused primarily on the immune cells themselves. Are there enough of them? Are the right subtypes present? Are the checkpoint molecules expressed at the right levels? Are there too many suppressive cell types in the tumor? These questions are not wrong. They are real and important. But they have implicitly treated the cancer cell's own biochemistry as a backdrop, a context for the immune response, rather than an active participant in shaping it. What the NRF2 data was suggesting was that the cancer cell's metabolic program, specifically its management of oxidative stress, was not merely a feature of cancer survival. It was a tool of immune evasion. Cancer cells with constitutive NRF2 activity were not just protecting themselves. They were, through the simple act of exporting their antioxidant excess, creating a microenvironment that disabled the immune response around them. remove that biochemical sabotage, and the immune system, at least in these models, did much of the therapeutic work itself. By the summer of 2024, the team had enough data to submit a comprehensive manuscript to Nature Cancer, one of the highest impact journals in the oncology field. The paper, which ran through peer review over approximately 4 months and required 2 rounds of revisions, documented the full arc of the research: the initial cell culture findings, the PDX chemotherapy resensitization data, the transcriptomic discovery of microenvironment remodeling, and the triple combination results in humanized models. The peer review process, characteristically rigorous for a journal of this standing, required the team to address several substantive scientific questions. Reviewers asked for additional validation of the off-target editing analysis using more sensitive next-generation sequencing methods. They requested clarification of the mechanistic pathway connecting NRF2 deletion to glutathione export and T-cell redox signaling, including direct measurement of extracellular glutathione concentrations in treated versus untreated tumors. And they pressed for a clearer presentation of the limitations of the humanized mouse model system and the caveats that apply when extrapolating these findings toward human clinical scenarios. The team provided all of this. The additional next-generation sequencing analysis, using a method called GuideSeq, which is considerably more sensitive for detecting off-target CRISPR edits than the standard approaches, confirmed that off-target editing in healthy tissues remained below the threshold of detection. The direct metabolomic measurements showed that extracellular glutathione concentrations in NRF2 edited tumors decreased by 63 to 71% compared to unedited controls, confirming the mechanistic hypothesis. And the discussion section of the final manuscript was notably careful in its language, explicitly delineating what the mouse data could and could not be taken to predict about human clinical outcomes. The paper was accepted in November 2024 and published in Nature Cancer in the December 2024 issue. Its title was Measured: CRISPR-mediated NRF2 disruption resensitizes chemotherapy-resistant non-small cell lung cancer and remodels the immunosuppressive tumor microenvironment. Peer-reviewed, independent, in the literature. The response from the scientific community was swift and substantive. Within weeks of publication, the paper had accumulated an unusual volume of citations for such a recent work, suggesting that researchers across multiple fields, cancer genomics, immuno-oncology, CRISPR therapeutics, metabolic oncology, were reading it carefully and incorporating its findings into their own research frameworks. Commentary pieces appeared in Nature Review's Cancer and Cancer Cell. Most were measured in their enthusiasm, appropriately so for findings that had not yet been replicated by independent groups and had not been tested in humans. But the consensus view, as expressed in these commentaries, was that the mechanistic story was coherent, the experimental design was rigorous, and the therapeutic hypothesis was worth pursuing in a clinical context. One commentary, authored by Dr. Johanna Olveus at the Oslo University Hospital and published as a correspondence piece in Nature Cancer in January 2025, put it succinctly. If the glutathione-mediated immune suppression mechanism described here translates to human tumors, it would represent one of the most actionable metabolic explanations for checkpoint inhibitor resistance discovered to date. That phrase, actionable, carries specific meaning in the oncology world. It means there is a clear therapeutic intervention that could address the mechanism. It means the biology points to a treatment. It means the path from discovery to patient, while still long and uncertain, is at least visible. But the path to patients is never simple. And in early 2025, even as the publication was generating scientific momentum, the team was confronting a set of practical challenges that no amount of elegant biology could resolve by itself. The CRISPR delivery system, the engineered extracellular vesicles, had worked in mice. But manufacturing extracellular vesicles at the quality, quantity, and consistency required for human clinical trials was a problem of an entirely different order of magnitude than producing enough for a small animal study. The regulatory pathway for a CRISPR-based therapy targeting a solid tumor in vivo, meaning the editing happening inside the patient's body rather than outside it in a laboratory, was largely uncharted territory. The Food and Drug Administration had issued guidance documents on gene therapy products, and CRISPR ex vivo therapies had been approved in specific contexts. But in vivo CRISPR editing in solid tumors remained in a regulatory gray zone that would require extensive preclinical toxicology data, careful investigational new drug application preparation, and close dialogue with the FDA's Center for Biologics Evaluation and Research. And there was the question of patient selection. Which lung cancer patients specifically would be candidates for this therapy? Only those with confirmed NRF2 pathway mutations? A broader population? How would tumors be screened? What companion diagnostic would be used? These questions were not scientific in the traditional sense. They were translational, the hard engineering work of turning a laboratory discovery into a clinical reality. And they required a different kind of expertise than the molecular biology that had produced the original findings. In the spring of 2025, the San Diego team announced a formal collaboration with a clinical research group at the University of Texas MD Anderson Cancer Center, specifically the Department of Thoracic Oncology, headed by one of the field's most experienced lung cancer clinicians. The collaboration would be focused on designing a Phase 1 clinical trial, the first in-human safety study that, if regulatory approval could be obtained, would begin the process of determining whether any of what the mice had shown could be reproduced in the patients who needed it most. That process, the design of the trial, the regulatory dialogue, the manufacturing challenges, and the first cautious steps toward human testing, is a story still being written at the time of this video. Phase 1 trial design documents were under preparation through late 2025. The timeline, as stated by the research team in interviews published through early 2026, targets an investigational new drug application filing with the FDA in the first half of 2026. No human patients have yet been treated. That must be stated clearly. Everything described so far, as remarkable as it is, remains at the preclinical stage. But what has been established is real, and what it suggests about the biology of lung cancer resistance is worth pausing on before this investigation moves forward. The bigger picture. In February of 2025, a clinical data review meeting took place in a conference room at the European Institute of Oncology in Milan, Italy. These meetings happen constantly in cancer research: routine, administrative, procedural. Committees of clinicians and biostatisticians gather around tables covered with printouts and laptop screens, reviewing trial data, checking safety parameters, making incremental decisions about ongoing studies. This particular meeting was reviewing interim data from a Phase 2 clinical trial of pembrolizumab, the checkpoint inhibitor marketed under the brand name Keytruda by Merck, in patients with advanced non-small cell lung cancer who had previously failed platinum-based chemotherapy. The trial was not unusual in its design. Checkpoint inhibitor trials in previously treated lung cancer were commonplace by 2025, representing one of the most active areas of clinical investigation in all of oncology. What was unusual was what one of the biostatisticians noticed when she stratified the response data by molecular subtype. Her name was Dr. Francesca Mori, and she had been analyzing clinical trial data at the European Institute for 11 years. She was not a basic scientist. She did not work with CRISPR or cell lines or mouse models. Her expertise was in the numbers, survival curves, hazard ratios, confidence intervals, the statistical architecture of clinical evidence. And the numbers she was looking at on that February morning were telling her something that the trial had not been designed to detect, but that emerged from the data regardless, with a statistical clarity she could not dismiss. The trial had enrolled 214 patients with previously treated non-small cell lung cancer, standard population for this kind of study. All patients had tumor samples available for molecular profiling, and as part of the correlative science embedded in the trial protocol, those samples had been analyzed for a panel of genomic alterations including, as a matter of routine rather than primary interest, mutations in the KEAP1 and NRF2 genes. 31 of the 214 patients, approximately 14.5%, carried mutations in either KEAP1 or NRF2 that were predicted to result in constitutive NRF2 pathway activation. This was consistent with the frequencies reported in the literature. When Dr. Mori stratified the pembrolizumab response data by KEAP1 NRF2 mutation status, the separation between the two groups was stark. In patients without KEAP1 NRF2 pathway mutations, the objective response rate to pembrolizumab, meaning the fraction of patients whose tumors shrank by at least 30% by standard measurement criteria, was 38%. Consistent with the broader literature on pembrolizumab in this setting. In patients with KEAP1 NRF2 pathway mutations, the objective response rate was 6.5%, 2 patients out of 31. And both of those two responses were partial and short-lived, with disease progression occurring within 4 months of the initial response. The median progression-free survival, the time until the disease worsened despite treatment, was 2 months in the KEAP1 NRF2 mutant group, compared to 6.4 months in the non-mutant group. 6 times lower response rate, 3 times shorter progression-free survival. In the same drug, the same disease, the same clinical setting, the only difference was the status of one regulatory axis, KEAP1 and NRF2. Dr. Mori brought the finding to her colleagues immediately. The trial's principal investigator, Dr. Alessandro Bettini, reviewed the analysis and recognized its significance within the context of the San Diego research that had been published 3 months earlier in Nature Cancer. He convened an emergency supplementary analysis that consumed the next 6 weeks of the biostatistics team's time. The supplementary analysis added depth to the initial finding in ways that made it more, rather than less, alarming. The team looked at overall survival, not just progression-free survival, but how long patients lived from the time of trial enrollment. In the KEAP1 NRF2 mutant group, median overall survival on pembrolizumab was 7.2 months. In the non-mutant group, median overall survival had not yet been reached at the time of the interim analysis, meaning that enough patients in that group were still alive that the median point had not been crossed. The two curves on the Kaplan-Meier survival plot, the standard graphical representation of survival data in oncology trials, diverged early and never converged. From the very beginning of treatment, the KEAP1 NRF2 mutant patients were following a fundamentally different and worse trajectory. Checkpoint inhibitor therapy was not just working less well in this group, it appeared to be providing almost no meaningful benefit at all. And here is the detail that transformed this clinical observation from a statistical footnote into a therapeutic emergency. These were patients who had already failed platinum-based chemotherapy. They had already proven resistant to the standard first-line treatment. Pembrolizumab was, for most of them, what oncologists call the second-line option, the treatment of next resort after the first line. If pembrolizumab was also failing in this subgroup, failing dramatically and consistently, then these patients were arriving at an impasse with essentially no validated systemic treatment options remaining. 14.5% of non-small cell lung cancer patients. In the United States alone, according to data from the National Cancer Institute's Surveillance, Epidemiology, and End Results Program, approximately 234,000 new cases of lung cancer are diagnosed annually, with non-small cell variants accounting for roughly 80 to 85% of that total. 14.5% of those patients, doing the arithmetic carefully, represent somewhere in the range of 27,000 to 30,000 people in the United States every year who may carry KEAP1-NRF2 pathway mutations. 27,000 to 30,000 people a year in one country for whom both standard chemotherapy and checkpoint immunotherapy were delivering sharply diminished benefit. Globally, given the worldwide lung cancer incidence figures, the number would scale to hundreds of thousands. These were not abstractions. These were patients sitting in oncology clinics, receiving treatments that the emerging data strongly suggested were not working for their specific tumor biology. Dr. Bettini submitted the supplementary analysis as a rapid communication to the Journal of Clinical Oncology, where it was published in May 2025 under the title, KEAP1-NRF2 Pathway Mutations as a Predictor of Checkpoint Inhibitor Resistance in Previously Treated Non-Small Cell Lung Cancer, Interim Analysis of a Phase 2 Trial. The publication triggered an immediate and substantive response from the oncology community that went well beyond the customary academic exchange of letters and commentaries. Within weeks of the JCO publication, the European Society for Medical Oncology issued a clinical alert, not a guideline change, which takes considerably longer and requires a higher standard of evidence, but a formal communication to member oncologists, drawing attention to the finding and recommending that clinicians consider KEAP1-NRF2 mutation status as a factor in treatment discussions with patients who had advanced non-small cell lung cancer. The American Society of Clinical Oncology convened a working group to review the emerging data and assess whether updated practice guidance was warranted. This process, as of early 2026, was ongoing, and several major cancer centers, including Memorial Sloan Kettering in New York, the MD Anderson Cancer Center in Houston, and the Christie Hospital in Manchester, UK, began incorporating KEAP1-NRF2 pathway mutation testing into their standard molecular profiling panels for lung cancer patients, going beyond what was required by existing guidelines. The clinical response was measured but serious. The scientific community was not prepared to overturn treatment paradigms based on interim data from a single Phase 2 trial. The evidence for the checkpoint inhibitor resistance association was compelling but preliminary. Independent validation was needed. larger data sets, prospective rather than retrospective molecular stratification. But the clinical direction was clear enough that practicing oncologists were beginning to factor this biology into their conversations with patients. For a scientific finding that had begun in a cell culture dish in San Diego in 2021, that was a remarkably rapid journey toward clinical relevance. Meanwhile, 3,000 miles from Milan, another thread of the NRF2 story was developing in a direction that neither the San Diego team nor the European clinical researchers had anticipated. At the Broad Institute of MIT and Harvard, the sprawling Genomics Research Center in Cambridge, Massachusetts, that has been one of the most productive engines of cancer biology discovery in the past 2 decades, a computational biology team led by Dr. Ramin Berokim had been conducting a different kind of analysis of NRF2's role in cancer. The Broad Institute's approach to cancer genomics operates at a scale that individual laboratories cannot match. Using datasets drawn from thousands of cancer cell lines, patient tumor samples, and functional genomic screens, Broad researchers can ask questions about cancer biology across the full diversity of human tumors in ways that smaller studies cannot access. The question Berokim's team was asking, beginning in late 2023, was whether NRF2 pathway mutations correlated not just with drug resistance, which was well established, but with specific patterns of tumor evolution. Specifically, did tumors with constitutive NRF2 activity accumulate genetic mutations at different rates than tumors with normal NRF2 regulation? The answer, published in a preprint posted to bioRxiv in September 2024 and subsequently submitted for peer review at Cancer Cell, was yes. And the mechanism was one that connected NRF2 biology to a process that oncologists had been trying to exploit therapeutically for years. Tumors evolve by accumulating mutations. This is simultaneously the source of their danger. Mutations drive resistance, and their potential vulnerability. Tumors with very high mutation burdens, called tumor mutational burden high, or TMB high tumors, generate large numbers of neoantigens, the abnormal protein fragments on the surface of cancer cells that immune cells can potentially recognize. High neoantigen load, in principle, gives the immune system more targets. This is part of the reason why TMB high tumors tend to respond better to checkpoint inhibitor therapy, and why some cancer types with notoriously high mutation rates, like melanoma, which accumulates UV radiation-induced mutations, and lung cancers in heavy smokers, were among the first to show impressive immunotherapy responses. But the relationship between TMB and immunotherapy response is imperfect. Some TMB high tumors respond poorly. Some TMB low tumors respond unexpectedly well. The predictive value of TMB alone, while real, is insufficient as a sole guide to treatment selection. What Berokim's team found was that NRF2 pathway status was interacting with tumor mutational burden in a way that had not been previously characterized. Tumors with constitutive NRF2 activation did accumulate mutations, but their mutation patterns were different from NRF2 normal tumors, with similar overall mutation burden. Specifically, NRF2 high tumors showed enrichment of a particular mutational signature, characterized by specific patterns of base substitution that is associated with defective DNA repair processes, rather than with the kind of immunogenic neoantigen-rich mutations that predict checkpoint inhibitor response. In plain language, NRF2 high tumors were accumulating mutations, but the wrong kind. They were generating genomic instability without generating the immunological visibility that genomic instability is supposed to produce. They were mutating in ways that made them genetically chaotic, but immunologically invisible. a combination that was simultaneously maximizing their evolutionary flexibility and minimizing their therapeutic vulnerability to immunotherapy. This was a finding with disturbing implications. It suggested that NRF2 pathway activation was not merely a passive driver of drug resistance. It was an active shaper of the tumor's evolutionary trajectory, influencing which kinds of mutations accumulated, and therefore what kind of tumor the cancer would become over time. Tumors with constitutive NRF2 activity were, in effect, evolving along a path that led toward greater resistance and reduced immune visibility simultaneously. The preprint generated considerable discussion in the cancer genomics community, particularly around the question of whether the observed mutation signature differences were a direct consequence of NRF2 activity or a result of confounding factors. The peer review process was ongoing as of early 2026, and the findings should be considered preliminary until formal publication and independent replication. But combined with the clinical data from the Milan trial, the picture emerging from the Broad Institute analysis reinforced a conclusion that the field was increasingly accepting. NRF2 was not a peripheral actor in lung cancer biology. It was a central one. There is a concept in oncology called the Cancer Hallmark Framework, introduced by Drs. Douglas Hanahan and Robert Weinberg in their landmark 2022 update to their classic paper originally published in Cell in 2000. The hallmarks describe the fundamental biological capabilities that cells must acquire to become cancerously malignant: sustaining proliferative signaling, resisting cell death, activating invasion and metastasis, inducing angiogenesis, reprogramming energy metabolism, avoiding immune destruction, and several others. Most individual oncogenes, the genes that drive cancer when mutated, contribute to one or two of these hallmarks. KRAS mutations, which are among the most common driver mutations in lung cancer, primarily drive proliferative signaling. TP53 mutations, which are present in roughly half of all human cancers, primarily disable cell death resistance. EGFR mutations, another major lung cancer driver, sustain growth factor signaling. NRF2 was emerging from the accumulating data as something qualitatively different. It was touching multiple hallmarks simultaneously: resistance to cell death through its antioxidant protection and apoptosis suppression, immune evasion through its metabolic manipulation of the tumor microenvironment, enabling replicative immortality, potentially through its role in shaping mutation accumulation patterns, reprogramming energy metabolism through its effects on glutathione biosynthesis and the pentose phosphate pathway. One gene, multiple hallmarks. That was the profile not of a peripheral player, but of what cancer biologists call a master regulator, a molecule so deeply embedded in the cancer cell's survival strategy that disrupting it could, in principle, unravel multiple layers of malignancy simultaneously. The therapeutic implications of that profile were significant, but so were the practical challenges of exploiting it. And the most pressing of those challenges, the one that was consuming the most attention and resources among the research teams working in this space in 2025, was the challenge that had been present from the beginning: delivery. To understand why delivery remains the central unsolved problem of CRISPR solid tumor therapy, it helps to think about scale. The human body contains approximately 37 trillion cells. A solid tumor of 1 centimeter in diameter, roughly the size of a small grape, which is large enough to be detected by standard imaging, but small enough to be considered early stage, contains somewhere in the range of 100 million to 1 billion cells. A tumor of 3 centimeters, still potentially resectable in lung cancer, but representing more advanced local disease, may contain 10 billion cells or more. To functionally disrupt NRF2 throughout a tumor, you do not need to edit every cancer cell. The experiments in San Diego had shown effectiveness with editing efficiencies in the range of 60 to 80%. But 60% of 1 billion cells is still 600 million cells. And the CRISPR editing machinery needs to reach each of those cells individually, penetrate the cell membrane, navigate to the nucleus, find the specific target sequence, and make the cut. The extracellular vesicle delivery system that had shown promise in the PDX models was elegant in its targeting mechanism, but it was not yet a scalable clinical product. Extracellular vesicles are produced by living cells, typically through a process of cell culture followed by centrifugation to collect the secreted particles. The yield from standard cell culture processes is sufficient for mouse experiments, but orders of magnitude short of what would be required for clinical manufacturing. Pharmaceutical manufacturing of biological therapies is governed by standards of consistency, purity, potency, and sterility that are, for good reason, extraordinarily demanding. A therapy injected into a human patient must be produced with essentially zero batch-to-batch variability in the parameters that matter for safety and efficacy. A delivery vehicle for CRISPR components must be proven to carry its payload consistently, reach its target consistently, and release the editing machinery consistently across every vial in every batch produced. Achieving those standards with extracellular vesicles, which are inherently heterogeneous biological particles, varying in size, surface composition, and cargo loading from one production run to the next, was a manufacturing challenge that no team in the world had yet fully solved as of early 2026. Several biotechnology companies were working on the problem. Evox Therapeutics, based in Oxford, UK, had been developing engineered extracellular vesicle platforms for therapeutic payload delivery since its founding in 2016, and had published preclinical data on exosome-mediated delivery of nucleic acid therapeutics across multiple disease areas. Kodiak Biosciences, a Cambridge, Massachusetts-based company, had conducted the first clinical trial of an engineered exosome therapeutic, a different application, not CRISPR-based, before pivoting its business model in 2023. Several other companies in the United States, Europe, and Asia had active programs in the space. None had yet demonstrated clinical-grade manufacturing of CRISPR-loaded extracellular vesicles at the scale and consistency required for a human efficacy trial in solid tumors. This was not a reason for pessimism. It was a reason for sober realism about timelines. The history of biological therapeutics is filled with examples of molecules that worked beautifully in the laboratory and required years of manufacturing innovation before they could reach patients. Monoclonal antibodies, now the backbone of immunotherapy and one of the most successful drug classes in pharmaceutical history, faced manufacturing challenges in their early development that at the time seemed nearly insurmountable. They were surmounted because the therapeutic promise was real and the investment followed. The same logic applied here. If the biology continued to hold, if the NRF2 story translated from mice to humans, the manufacturing problem would be solved. Not easily, not quickly, but inevitably. Because the therapeutic target was real, the mechanism was understood, and the clinical need was acute. There was, however, an alternative delivery approach that was attracting significant attention in parallel to the extracellular vesicle And this approach came from an unexpected direction, from the very technology that had already demonstrated its clinical viability at massive scale in a completely different medical context. Lipid nanoparticles, the delivery vehicles of the COVID-19 mRNA vaccines. The success of the Moderna and Pfizer-BioNTech COVID vaccines had done something beyond protecting hundreds of millions of people from a respiratory virus. It had transformed lipid nanoparticle technology from a laboratory curiosity into a clinically validated, manufacturing-ready platform. The infrastructure for producing lipid nanoparticles at pharmaceutical scale, the equipment, the expertise, the regulatory precedent, now existed in a way it had not before 2020. The challenge of using lipid nanoparticles for tumor-targeted CRISPR delivery was the same challenge that had always limited their application to solid tumors. Lipid nanoparticles, when administered intravenously, accumulate preferentially in the liver. This is determined by the absorption of specific blood proteins onto the nanoparticle surface, which routes them to liver cells through receptor-mediated mechanisms. But by 2024 and 2025, a new generation of surface-modified lipid nanoparticles, engineered with targeting ligands that redirected their organ tropism away from the liver and toward specific cell types, was showing early promise in preclinical models. Research groups at MIT, at the University of Pennsylvania, and at ETH Zurich published data on ionizable lipid formulations that could be tuned, through modification of their lipid composition and surface chemistry, to accumulate in the lungs, in the spleen, or in specific tumor types expressing targeted. A collaboration between the University of Pennsylvania's Lipid Nanoparticle Research Group and the San Diego team was formalized in mid-2025, combining the San Diego researchers' expertise in NRF2 biology and CRISPR guide RNA design with Pennsylvania's lipid nanoparticle engineering platform. The goal was to develop a parallel delivery approach to the extracellular vesicle system, not to replace it, but to provide an alternative pathway to the clinic if the manufacturing challenges of the extracellular vesicle system proved too slow to overcome. Two parallel delivery strategies targeting the same gene, designed to reach the same tumor. In the history of cancer drug development, having redundancy in the approach, multiple paths toward the same therapeutic goal, is not a luxury, but a necessity. Most experimental therapies fail, not because the target is wrong, but because of delivery, manufacturing, toxicology, or regulatory obstacles that could not be anticipated from preclinical data alone. The teams in San Diego were not naive about this. They had read enough drug development history to know that the road from compelling preclinical data to an approved cancer therapy is littered with promising findings that never made it to patients. They were building redundancy deliberately. They were hedging scientifically, because scientific hedging is not weakness, it is wisdom. And running in parallel to all of this translational engineering work was a scientific question that had not left the back of Dr. Chen's thinking since the autumn 2023 analysis that had first revealed NRF2's pan-cancer scope. If NRF2 was a master regulator touching multiple cancer hallmarks in lung cancer, and if the same pathway was dysregulated in liver cancer, head and neck cancer, bladder cancer, esophageal cancer, what would happen if you asked the CRISPR NRF2 deletion strategy in those other cancer types? Experiments were underway. In cell lines, in PDX models, in collaboration with researchers at MD Anderson who were establishing NRF2 mutant models of hepatocellular carcinoma. The data, as of early 2026, was accumulating but not yet published. But the direction of the preliminary results shared at an internal consortium meeting in January 2026, according to a summary description provided in a grant report made publicly available through the National Institutes of Health Reporter database, was described as consistent with the lung cancer findings across all tumor types tested to date. Consistent across all tumor types tested. That phrase, in the careful understatement of scientific reporting, carries a weight that deserves recognition. It means that what began as a lung cancer story may be telling a broader truth about cancer itself, about how tumors across diverse tissues and molecular backgrounds use NRF2 as a common currency of survival, and how disrupting that currency might, in principle, restore the effectiveness of treatments that cancer had learned to evade. But before this investigation turns to the question of what all this means for the future of cancer treatment, a question that demands both ambition and careful honesty about what is proven versus what is possible, there is one more dimension of the NRF2 story that has not yet been fully explored. Because NRF2 is not only a therapeutic target, it is also a diagnostic. And the emerging data on NRF2 pathway mutation testing in clinical practice, on who should be tested, how they should be tested, and what the results should mean for treatment selection, opens a chapter of the story that is as important for patients today as any future CRISPR therapy might be for patients tomorrow. You do not need to wait for gene editing to reach the clinic to use what NRF2 biology has already revealed. In some cancer centers, quietly and without headlines, the knowledge accumulated in laboratories over the past several years is already changing how patients with non-small cell lung cancer are being evaluated, stratified, and treated. And that part of the story, the part about what patients and oncologists can do right now with the knowledge already in hand, is where the next chapter begins. What this means for patients today. There is a particular cruelty embedded in the gap between scientific discovery and clinical practice. A researcher publishes a finding that could change how a disease is treated. The evidence accumulates, the consensus shifts, and somewhere in an oncology clinic in Ohio or Lyon or Osaka, a patient sits across from a physician who has not yet incorporated that knowledge into the conversation. Not because the physician is negligent, but because the machinery of medical translation moves slowly, unevenly, and with enormous variation between institutions. The NRF2 story is, right now, in that gap. The science has moved faster than the clinical infrastructure has caught up. And that means that the information in this chapter about what patients with non-small cell lung cancer can ask for today, what tests exist, what the results might mean for treatment decisions, is not hypothetical. It is immediately actionable. And for some patients, it may be the difference between receiving a treatment that works for their specific tumor biology and receiving a treatment that the emerging evidence strongly suggests will not. Let us be precise about what that means in practice. The fundamental clinical question raised by everything discussed in the previous chapters is straightforward to state, even if the institutional response has been uneven. For a patient diagnosed with non-small cell lung cancer, particularly a patient with advanced or metastatic disease who is being evaluated for systemic treatment, what is the NRF2 pathway status of their tumor? This is not a futuristic question requiring experimental technology. It is a question that can be answered today using tools that are already available in major cancer centers. Standard comprehensive genomic profiling of lung cancer tumors, the kind of next-generation sequencing panel that analyzes hundreds of cancer-relevant genes simultaneously from a tumor biopsy sample, already includes KEAP1 and NRF2 in its gene panels. The most widely used commercial comprehensive genomic profiling tests in the United States, including the Foundation 1 CDX panel developed by Foundation Medicine, which received FDA approval as a companion diagnostic for multiple lung cancer therapies, includes both KEAP1 and NRF2 in its 324-gene panel. The test requires tumor tissue, either from a surgical resection sample, a core needle biopsy, or in some cases a liquid biopsy using circulating tumor DNA from a blood sample. Turnaround time from sample submission to result is typically 10 to 14 business days for tissue-based testing. The test is covered by Medicare for patients with advanced solid tumors who meet eligibility criteria, and coverage by commercial insurance varies, but has expanded significantly in recent years as comprehensive genomic profiling has moved from experimental to standard of care in advanced lung cancer. So the test exists. The genes are on the panel. The coverage pathway exists. And the clinical significance of the result is now supported by published data in peer-reviewed journals. The problem is not availability. The problem is interpretation and action. When a comprehensive genomic profiling report comes back with a KEAP1 loss-of-function mutation or an NRF2 gain-of-function mutation identified in the tumor, what should happen next? Until very recently, the answer in most oncology practices was not much. KEAP1 and NRF2 mutations were noted in the genomic report, classified as variants of uncertain or known significance depending on the specific alteration, and then largely set aside because there was no approved targeted therapy that acted directly on this pathway. The mutations were documented but not actionable in the therapeutic sense. The research from 2024 and 2025 has begun to change that calculus. Not by providing an approved targeted therapy, there is none yet, but by providing evidence that KEAP1 NRF2 mutation status should inform treatment sequencing decisions even within existing treatment options. Specifically, the Milan trial data, published in the Journal of Clinical Oncology in May 2025, provides the most clinically direct evidence to date that KEAP1 NRF2 pathway mutations are associated with dramatically reduced benefit from pembrolizumab monotherapy in previously treated non-small cell lung cancer. An objective response rate of 6.5% versus 38% is not a marginal difference. It is a difference that, if confirmed in larger prospective studies, would have direct implications for which patients should be offered pembrolizumab as their primary second-line option and which patients might be better served by alternative approaches. What are those alternative approaches? At the current level of evidence, the answers are limited, and honesty about those limits is essential. For patients with KEAP1 NRF2 mutant non-small cell lung cancer who have progressed on first-line platinum-based chemotherapy, the standard second-line options at major cancer centers currently include single-agent docetaxel, combination docetaxel plus ramucirumab, an anti-angiogenic antibody, and pembrolizumab, or other checkpoint inhibitors. All of these options have modest efficacy in the second-line setting for unselected populations. The NRF2 pathway data suggests that pembrolizumab may be particularly poorly suited for the NRF2 mutant subgroup, while the data on docetaxel-based regimens in this specific molecular context is less well characterized. Several oncologists at major cancer centers who have incorporated NRF2 pathway mutation testing into their practice have described, in conference presentations and published correspondence, a clinical approach of offering KEAP1 NRF2 mutant patients enrollment in clinical trials as a preferred option over standard second-line therapy, on the rationale that the expected benefit from standard options is low enough that the potential benefit of experimental approaches justifies the uncertainties of trial participation. This is a nuanced clinical judgment, not a guideline-endorsed recommendation. But it reflects a genuine and growing recognition that KEAP1 NRF2 mutant lung cancer is a molecularly distinct disease entity that may require molecularly tailored therapeutic strategies. For patients in the first-line setting, those being evaluated for initial systemic treatment after diagnosis with advanced non-small cell lung cancer, the implications of NRF2 pathway mutation status are different and perhaps more immediately important. Current standard first-line treatment for advanced non-small cell lung cancer without actionable driver mutations, meaning without EGFR mutations, ALK rearrangements, ROS1 rearrangements, or other targeted therapy-eligible alterations, is typically pembrolizumab-based, either as monotherapy for patients with high PD-L1 expression or in combination with platinum-based chemotherapy for patients with lower PD-L1 expression. The combination of pembrolizumab plus platinum chemotherapy is a standard of care endorsed by major oncology guidelines, and the evidence supporting it in unselected populations is robust. But the NRF2 pathway data raises a question that has not yet been prospectively addressed in a large clinical trial. Does KEAP1 NRF2 mutation status predict differential benefit from pembrolizumab-containing first-line combinations compared to chemotherapy alone? Retrospective analyses from large clinical trial databases have begun to address this question. A pooled analysis of data from 3 major pembrolizumab plus chemotherapy trials in non-small cell lung cancer, presented at the European Society for Medical Oncology annual meeting in September 2025 by researchers from the National Cancer Institute and the Dana-Farber Cancer Institute, examined outcomes stratified by KEAP1 mutation status in patients for whom archival genomic data was available. The results, which have been submitted for peer review and should be considered preliminary until published, suggested a pattern consistent with the Milan trial data. Patients with KEAP1 loss mutations appeared to derive less benefit from the addition of pembrolizumab to chemotherapy compared to KEAP1 intact patients, though the magnitude of the difference was smaller in the first-line combination setting than in the second-line monotherapy setting. The chemotherapy backbone may partially offset the immunotherapy resistance driven by NRF2 pathway activation, though it does not eliminate it. Again, these are preliminary findings from retrospective analyses. They are not sufficient to change guidelines or to recommend withholding pembrolizumab from KEAP1 NRF2 mutant patients in clinical practice. But they are sufficient to motivate the design of prospective trials specifically enrolling KEAP1 NRF2 mutant patients, trials that are now in various stages of design and regulatory preparation at multiple institutions. Beyond treatment selection, there is a second clinical dimension of NRF2 pathway biology that deserves attention: its implications for monitoring treatment response. One of the persistent challenges of cancer treatment is knowing, in real time and without waiting for imaging confirmation, whether a treatment is working. Imaging, CT scans, PET scans, provides the most direct assessment of tumor response. but scans are typically performed every 6 to 12 weeks at minimum, and each scan involves radiation exposure, cost, and patient burden. Liquid biopsy, the analysis of tumor-derived DNA circulating in the bloodstream, has emerged as a powerful tool for monitoring treatment response in real time. Tumors shed small fragments of their DNA into the blood continuously. As a tumor shrinks in response to treatment, the amount of circulating tumor DNA in the blood decreases. As a tumor becomes resistant and begins to grow again, circulating tumor DNA levels rise, often weeks before the growth becomes visible on imaging. For KEAP1 NRF2 mutant lung cancers specifically, liquid biopsy offers a potentially important monitoring tool. Because the KEAP1 or NRF2 mutation is specific to the tumor, it is not present in the patient's normal cells. It serves as a tumor-specific molecular marker that can be tracked in blood samples. Rising or falling levels of the KEAP1 or NRF2 mutant allele fraction in circulating tumor DNA can, in principle, provide an early read on treatment response or resistance emergence. Several cancer centers, including Memorial Sloan Kettering and MD Anderson, have published case series and small cohort analyses describing the use of serial liquid biopsy monitoring in KEAP1 mutant lung cancer patients, with circulating tumor DNA dynamics correlating with clinical outcomes in the majority of reported cases. The evidence base is currently too small for this approach to be considered standard clinical practice, but liquid biopsy monitoring in genomically defined lung cancer subgroups is one of the most active areas of clinical research in thoracic oncology, and the data is accumulating rapidly. The question of clinical trial access deserves special emphasis because it represents the most direct pathway through which patients with KEAP1 NRF2 mutant lung cancer can access therapies that go beyond the current standard of care. As of early 2026, several clinical trials relevant to NRF2 pathway biology in lung cancer are actively enrolling or in final stages of design. At the MD Anderson Cancer Center, a Phase 1-2 trial evaluating a combination of the MEK inhibitor trametinib, which has been proposed as a potential synthetic lethal partner for NRF2 high tumors based on preclinical data, with docetaxel in patients with previously treated KRAS mutant non-small cell lung cancer enriched for KEAP1 co-mutations, is actively enrolling. KRAS and KEAP1 mutations frequently co-occur in lung adenocarcinoma, and preclinical data suggests that this combination may have particular activity in the NRF2 high context. The trial identifier in the clinicaltrials.gov database is NCT 054-18379. At Memorial Sloan Kettering, a biomarker-selected trial evaluating whether KEAP1NRF2 mutation status predicts differential benefit from standard first-line treatment combinations is in late-stage design, with enrollment anticipated to open in mid-2026, pending final regulatory review. In Europe, the ETOP-LUNG International Consortium, a cooperative group coordinating academic clinical trials in thoracic oncology across European centers, announced in November 2025 the initiation of protocol development for a randomized Phase 2 trial in KEAP NRF2 mutant non-small cell lung cancer, comparing standard pembrolizumab-based therapy to an experimental arm incorporating NRF2 pathway-targeted intervention. The specific experimental arm design had not been publicly disclosed as of early 2026, pending ongoing preclinical validation and regulatory consultation. And the Phase 1 trial, being designed by the San Diego team in collaboration with MD Anderson, the first in-human safety study of CRISPR-mediated NRF2 editing, remained on track, according to statements from the research team, for investigational new drug application filing with the FDA in the first half of 2026. If the IND is approved and the manufacturing challenges of the delivery system can be addressed on schedule, first patient enrollment could potentially occur in 2027, though the team has been careful to note that these timelines are subject to the many variables that govern clinical trial initiation. For patients interested in clinical trial options, the clinicaltrials.gov database, maintained by the National Library of Medicine, is the most comprehensive, publicly accessible resource for identifying open trials by cancer type, molecular profile, and geographic location. Searching for KEAP1 or NRF2 alongside non-small cell lung cancer in that database returns the current landscape of relevant trials and their enrollment criteria. There is another dimension of the Today story that tends to get lost in the excitement of molecular discovery. The question of what patients should be asking their oncologists right now, in practical terms, at their next appointment. The answer is simpler than the science that underlies it. If you have been diagnosed with non-small cell lung cancer, ask your oncologist whether comprehensive genomic profiling of your tumor has been performed. This is now standard of care in advanced non-small cell lung cancer and should be offered to all patients with metastatic disease at the time of initial diagnosis. If it has not been done, ask why and request referral to a center that routinely performs it. If comprehensive genomic profiling has been performed, ask specifically whether your report included results for KEAP1 and NRF2. If mutations in either gene were identified, ask what those mutations mean for the expected response to pembrolizumab-based therapy, and whether clinical trial options specific to your molecular profile are available. If you are at a community oncology center rather than a major academic cancer center, consider requesting a second opinion consultation at a National Cancer Institute designated comprehensive cancer center, where molecular tumor boards, multidisciplinary expert committees that review complex genomic profiling results and provide treatment recommendations, are more consistently available. NCI-designated comprehensive cancer centers are listed on the National Cancer Institute website and exist in all regions of the United States. These are not extraordinary requests. They are precisely the kind of informed, molecularly guided questions that the oncology community has been encouraging patients to ask for years. The NRF2 story has simply provided new and specific scientific content to fill those questions. There is one more aspect of the Today picture that deserves honest discussion, because it involves a tension that runs through all of precision oncology and is particularly acute in the NRF2 case. The tension is between the growing sophistication of molecular diagnosis and the much slower pace of molecular therapeutics. Comprehensive genomic profiling can now identify KEAP1 NRF2 pathway mutations with high sensitivity and specificity. The clinical significance of those mutations is increasingly well characterized, but the therapeutic options specifically designed to address those mutations remain, as of early 2026, either in preclinical development or in early clinical trial stages. This creates a situation that oncologists find genuinely difficult. You can tell a patient precisely what molecular feature is making their cancer resistant to available treatments, but you cannot yet offer them a validated treatment that addresses that specific feature. The diagnostic capability has outrun the therapeutic capability. This is not unique to NRF2. It is a recurring pattern in precision oncology. The era of comprehensive genomic profiling has identified dozens of potentially actionable alterations across many cancer types for which matched therapies are either unavailable or still experimental. Patients receive molecular reports that are rich with information and sometimes poor in immediately applicable clinical implications. What this means in practice is that molecular knowledge, right now, is most valuable not as a direct therapeutic guide, but as a tool for informed decision-making about treatment sequencing, clinical trial eligibility, and the allocation of treatment intensity. Knowing that a patient's tumor has constitutive NRF2 activation may not point to a specific approved drug, but it may point away from a treatment, pembrolizumab monotherapy, that the evidence suggests is unlikely to work. And it may point toward a clinical trial that, while experimental, offers the most scientifically rational approach to their specific tumor biology. In medicine, sometimes the most valuable thing knowledge can do is tell you what not to do. And in the NRF2 case, the emerging evidence is already doing that in cancer centers sophisticated enough to apply it. There is a human story behind every statistical abstraction in this chapter, and it is worth pausing on one of them. Not a specific named patient. Patient privacy is an ethical boundary that clinical reporting appropriately observes. But a composite portrait drawn from the kinds of cases described in published case series and clinical conference presentations. Imagine a 62-year-old woman, a former smoker, diagnosed 18 months ago with stage 4 lung adenocarcinoma. Her tumor was profiled at diagnosis, revealing a KRAS G12C mutation, a targetable driver, alongside a co-occurring KEAP1 loss-of-function mutation. She was treated initially with sotorasib, the first approved KRAS G12C inhibitor, and responded well for 7 months. Then her disease progressed. At progression, her oncologist faced a choice between pembrolizumab monotherapy, her PD-L1 expression was 40%, in the range that would normally make pembrolizumab a reasonable option, and enrollment in a clinical trial specifically enrolling KEAP1 mutant patients who had progressed on KRAS-targeted therapy. The oncologist, at a major cancer center with an active molecular tumor board, brought the case to the board. The board reviewed the NRF2 literature, noted the Milan trial data suggesting limited pembrolizumab benefit in KEAP1 mutant tumors, and recommended trial enrollment as the preferred option over pembrolizumab monotherapy. The patient enrolled in a trial. Her outcome remains uncertain. Clinical trials, by their nature, do not guarantee benefit. But the decision was molecularly informed. It was made with knowledge rather than without it. And the difference between a decision made with molecular knowledge and one made without it is precisely the gap that the research described in this investigation is working to close. Not yet for everyone, not yet with a validated, targeted therapy. But in the clinics that are paying attention, and for the patients who know to ask, the knowledge is already making its way from the laboratory to the examining room. The skeptics, the competitors, and the hard questions. Science is not a chorus of agreement. It is an argument, a structured, evidence-governed, perpetually unresolved argument between people who have spent their careers thinking carefully about hard problems, and who disagree, sometimes profoundly, about what the evidence means and where it leads. The NRF2 story is no exception. For every researcher who reads the San Diego data and sees a transformative therapeutic opportunity, there is another who reads the same data and reaches for a list of objections. And those objections are not the reflexive resistance of entrenched thinking. Many of them are scientifically serious. They deserve to be heard with the same attention given to the findings themselves. Because the history of cancer research is, in significant part, a history of promising preclinical discoveries that did not survive contact with human biology. The graveyard of cancer therapies that worked brilliantly in mice and failed in humans is enormous. It contains molecules that cured tumors in rodents and produced no meaningful benefit in patients. It contains mechanistic hypotheses that were scientifically elegant and clinically irrelevant. It contains phase 3 trial failures that came after years of investment and genuine scientific excitement. None of this means the NRF2 story is wrong. It means the NRF2 story has not yet been tested in the crucible that ultimately matters, the human clinical trial. And until it has, the objections of careful skeptics are not obstacles to progress. They are the immune system of science itself. The first and most fundamental skeptical argument concerns the translational gap, the biological differences between mouse models and human patients that have undermined so many promising preclinical discoveries. The patient-derived xenograft models used in the San Diego experiments are, as noted earlier, more informative than simple cell line studies. They preserve tumor heterogeneity, three-dimensional architecture, and many aspects of the tumor microenvironment. But they are not human patients. PDX models are established in immunocompromised mice, animals whose immune systems have been partially or completely disabled to prevent rejection of the human tumor tissue. Even the humanized PDX models used in the combination immunotherapy experiments carry reconstituted human immune components that are imperfect representations of the complex, fully intact immune systems of cancer patients. The tumor microenvironment interactions that produced such striking results in those humanized models, the T-cell reactivation, the restoration of immune infiltration, the synergy with checkpoint inhibitor therapy, depend on a web of cellular and molecular interactions that no mouse model fully captures. Human tumors exist in the context of intact nervous systems, hormonal environments, microbiomes, aging immune systems with decades of exposure history, and metabolic states shaped by diet, medication, and chronic disease. None of these contextual factors are present in a mouse model. Dr. Catherine Arbour, a thoracic oncologist at Memorial Sloan Kettering Cancer Center, who has written extensively on KEAP1 NRF2 biology in clinical lung cancer, expressed this concern directly in a commentary published in the Journal of Thoracic Oncology in early 2020. She noted that while the mechanistic rationale for NRF2 targeted therapy was compelling, The history of immune microenvironment manipulation in preclinical models was littered with findings that did not translate. Therapies that remodeled the mouse tumor immune environment dramatically and produced minimal immunological benefit in human trials. The gap between showing T-cell reactivation in a humanized PDX model and demonstrating meaningful immune-mediated tumor control in a patient with advanced lung cancer and a complex treatment history is larger than the data currently bridges, she wrote. We should pursue this hypothesis vigorously. We should also pursue it with eyes open about what preclinical models can and cannot predict. This is not a dismissal of the research. It is a calibration of enthusiasm, precisely the kind of calibration that should accompany any preclinical finding before it reaches the clinic. The second major category of skeptical concern addresses the safety profile of NRF2 deletion specifically, and it is, in some ways, the most biologically complicated objection. NRF2 is not just a cancer gene. As described earlier, it is an essential regulator of cellular stress responses in healthy tissues. And among the healthy tissues that depend most critically on NRF2 function are several that are already under significant stress in lung cancer patients receiving aggressive treatment. The liver processes chemotherapy drugs and their metabolites. NRF2 is essential for hepatic detoxification responses. A patient receiving cisplatin or carboplatin alongside a CRISPR intervention that suppresses NRF2 in the liver could theoretically experience enhanced hepatotoxicity, drug-induced liver damage, because the liver's NRF2-dependent protective response has been impaired. The kidneys, which are the primary route of platinum drug elimination, are similarly dependent on NRF2 for protection against the nephrotoxicity that is one of the most clinically significant adverse effects of platinum-based chemotherapy. NRF2 activation in renal tubular cells limits cisplatin-induced kidney damage. Suppress NRF2 in the kidney, and that protection disappears. The lung itself, the very organ where the cancer lives, contains healthy epithelial cells, endothelial cells, and immune cells that rely on NRF2 for protection against oxidative injury. A patient with lung cancer already has compromised pulmonary function in many cases. Impaired NRF2 activity in the healthy lung tissue surrounding the tumor could, in principle, exacerbate respiratory compromise. The San Diego team's off-target editing data, showing that the extracellular vesicle delivery system achieved preferential editing in tumor tissue with minimal detectable editing in liver, kidney, and adjacent healthy lung tissue in the PDX models, directly addresses this concern. But the detection limit of the assays used means that very low levels of off-target editing cannot be excluded. And in a human patient receiving chemotherapy, with a liver and kidneys already metabolically stressed, even small amounts of NRF2 suppression in those organs could have consequences that were not apparent in the mouse models. Dr. Harmeet Mali, a hepatologist at the Mayo Clinic, who has studied NRF2 biology in the context of drug-induced liver injury, raised exactly this concern in a letter published in Nature Cancer in February 2025 in response to the December 2024. She called for explicit hepatotoxicity and nephrotoxicity monitoring protocols to be incorporated into the design of any first-in-human trial of NRF2 targeting CRISPR therapy, and for preclinical toxicology studies specifically designed to assess NRF2 suppression in the liver and kidney under conditions of concurrent platinum chemotherapy exposure. The San Diego team's response, published in the same issue, acknowledged these concerns as legitimate and stated that comprehensive organ toxicology studies were a central component of the preclinical development program underway in preparation for the investigational new drug application. They noted that the Phase 1 trial design would include intensive hepatic and renal function monitoring with predefined stopping rules if toxicity signals emerged. This exchange, concern raised, response given, monitoring plan incorporated, is science working as it should? The safety question is real. The commitment to addressing it rigorously is also real. And the Phase 1 trial, when it opens, will exist precisely to provide human safety data that no mouse model can generate. A third category of objection comes not from safety concerns, but from a fundamental question about therapeutic strategy. It is, in essence, an argument about whether CRISPR is the right tool for this particular job, even if the target is right. The argument goes like this. CRISPR-mediated gene editing is permanent. An edit made to a cancer cell is irreversible. The NRF2 gene in that cell is disrupted for the lifetime of the cell and all its progeny. In the context of killing cancer cells, this permanence is exactly what you want. You want the edit to be heritable, to propagate through tumor cell divisions, to ensure that resistance through NRF2 re-expression cannot emerge. But permanence is a double-edged problem. If the CRISPR delivery system is not perfectly specific, if even a small fraction of the editing machinery reaches healthy cells rather than tumor cells, those healthy cells carry a permanent disruption of a gene they need. A liver cell with a CRISPR-disrupted NRF2 gene cannot recover that gene when the therapeutic course ends. The damage is done. Pharmacological approaches, small molecule drugs that inhibit NRF2 protein activity rather than destroying the gene, are reversible. Stop administering the drug, and NRF2 activity recovers in healthy cells. This reversibility makes pharmacological approaches inherently safer in the event of off-target effects, because those effects resolve when treatment stops. The counter-argument, and it is a strong one, is that the pharmacological approach to NRF2 inhibition has been tried extensively and has consistently failed to produce specific, potent, and well-tolerated inhibitors. If the pharmacological approach had worked, we would not be having this conversation about CRISPR. The reason CRISPR is on the table is precisely because the small molecule approach has not delivered. But the pharmacological field has not stood still, and several of the most interesting competing approaches to the NRF2 problem are worth examining carefully, because they represent genuine scientific alternatives rather than retreads of failed strategies. At the University of Texas Southwestern Medical Center, a team led by Dr. Benjamin Tu has been pursuing an approach that targets NRF2 not directly, but through its metabolic dependencies. Their work, published in Cell Metabolism in June 2024, identified that NRF2 high cancer cells have a specific metabolic vulnerability, an unusual dependence on the amino acid cysteine for maintaining their elevated glutathione production. Cysteine is the rate-limiting precursor for glutathione synthesis. Cells cannot make glutathione without it. And NRF2 high cancer cells, which are producing glutathione at extraordinary rates to maintain their antioxidant excess, consume cysteine far faster than NRF2 normal cells. This creates a vulnerability. If you can deplete extracellular cysteine, the cysteine available for uptake from the tumor microenvironment, NRF2 high cancer cells run out of the raw material for their primary defense mechanism before normal cells do, because normal cells have much lower glutathione production demands. The tool for depleting extracellular cysteine is an enzyme called cystinase, an engineered protein that breaks down both cysteine and its oxidized form, cysteine, in the bloodstream and tumor microenvironment. Cystinase was originally developed at UT Southwestern by Dr. Everett Stone and had been studied in preclinical models of various cancers for several years before the 2 group demonstrated its particular potential in NRF2 high tumors. In cell culture and early mouse model experiments reported in the Cell Metabolism paper, cystinase treatment in combination with cisplatin produced synergistic cancer cell killing specifically in NRF2 high cancer cell lines, with substantially less effect on NRF2 normal lines, precisely the selectivity profile that a clinical candidate in this indication would need. The mechanism was direct. NRF2 high cells depleted their intracellular cysteine reserves faster when extracellular supply was cut off. Their glutathione production collapsed. Their antioxidant defenses failed, and cisplatin killed them. This approach has significant advantages over CRISPR in terms of clinical translatability. Cystinase is a protein, a biologic drug, that can be administered intravenously using well-established pharmaceutical manufacturing processes. It does not permanently alter any gene. It is reversible. And it does not require tumor-specific delivery. It acts in the extracellular environment of the tumor rather than inside cells, sidestepping the delivery challenge that haunts CRISPR solid tumor therapy entirely. The limitation is that the selectivity is not absolute. Normal cells also need cysteine, and systemic cysteine depletion would have effects on healthy tissues, particularly in tissues with high protein synthesis demands. The therapeutic window, the range of doses that deplete cysteine enough to harm NRF2 high cancer cells while preserving enough cysteine for critical healthy tissue functions, needs to be carefully characterized in toxicology studies. As of early 2026, cystinase for NRF2 high cancers was in advanced preclinical development, with Eterion Therapeutics, a company with licensing rights to the UT Southwestern technology, reporting that they were preparing for investigational new drug application filing. No human trials in lung cancer had opened as of this video. A second competing approach comes from the field of RNA therapeutics, specifically small interfering RNA or siRNA technology. siRNA works differently from CRISPR. Rather than cutting and disrupting the DNA gene itself, siRNA molecules bind to the messenger RNA produced from the NRF2 gene, the intermediate molecule that carries the genetic instructions from DNA to the protein-making machinery. When siRNA binds to NRF2 messenger RNA, it triggers the destruction of that messenger RNA before the NRF2 protein can be made. The result is reduced NRF2 protein levels, gene silencing rather than gene disruption. Like the pharmacological approach, siRNA silencing is not permanent. The siRNA molecules are degraded over time, and NRF2 messenger RNA production resumes. Multiple doses are required to maintain gene silence. This is simultaneously a limitation. You need repeated administration, and a safety advantage. You can stop treatment and NRF2 recovers. The delivery challenge for siRNA in solid tumors is similar to that for CRISPR. The molecules need to reach tumor cells specifically, avoid rapid degradation in the bloodstream, and penetrate cell membranes. Lipid nanoparticle delivery of siRNA has been clinically validated in the liver. The FDA-approved drug inclisiran uses this approach to silence PCSK9 in liver cells for cholesterol management. And patisiran, another lipid nanoparticle-delivered siRNA, has been approved for a rare genetic disease called transthyretin amyloidosis. For solid tumor delivery of siRNA beyond the liver, the challenges are similar to those facing CRISPR lipid nanoparticle delivery, and the solutions being pursued are conceptually parallel. Several biotech companies, including Cernomics and Alnylam Pharmaceuticals, have solid tumor siRNA delivery programs in various stages of preclinical development, though none specifically targeting NRF2 in lung cancer had entered clinical trials as of early 2026. A fourth competing approach represents perhaps the most intellectually heterodox position in the field: the argument that targeting NRF2 at all, in cancer, may be the wrong strategy. This argument comes primarily from a research group at the German Cancer Research Center in Heidelberg, Germany, led by Dr. Axel Ulrich. Their position, articulated in a perspective piece published in Cancer Research in November 2024, is based on a body of data showing that NRF2 has context-dependent effects in cancer, that it is not uniformly a driver of resistance and poor outcomes, but that in some tumor contexts, NRF2 activation is actually associated with better immune responses and improved checkpoint inhibitor sensitivity. The evidence cited includes analyses of specific cancer types, particularly certain subtypes of colorectal cancer and some melanoma subtypes, where high NRF2 activity correlates with increased immune infiltration rather than the immunosuppressive microenvironment described in the lung cancer studies. The German team argues that these context-dependent effects reflect fundamental differences in how NRF2 interacts with the immune microenvironment in different tumor types, shaped by the specific oncogenic context, the tissue of origin, and the co-occurring genetic alterations present in the tumor. Their conclusion is not that the San Diego findings are wrong. It is that the therapeutic implications of those findings may be specific to lung cancer and possibly to the KEAP1 NRF2 mutant subtype within lung cancer, and may not generalize to other tumor types without careful context-specific investigation. This is a scientifically substantive position, and it directly challenges the pan-cancer extrapolation that some in the field have been tempted to make from the San Diego and Broad Institute data. It argues for molecular contextualism, the idea that NRF2 is not simply good or bad for cancer, but that its role is determined by the specific biological environment in which it operates. The San Diego team has acknowledged this heterogeneity in their published work, and their preclinical expansion to other tumor types, the multi-cancer experiments underway as of early 2026, is explicitly designed to address which tumor contexts show NRF2 deletion benefit and which do not. The German group's concern is not with the lung cancer data, but with premature generalization. And premature generalization in cancer biology has a long history of causing wasted resources and misleading clinical decisions. Beyond the scientific debates, there is a category of hard questions surrounding CRISPR cancer therapy that falls outside the domain of molecular biology entirely. These are ethical, regulatory, and economic questions, and they are as important to the real-world outcome of this research as any experiment in a laboratory. The regulatory question is genuinely complex. In vivo CRISPR editing, making permanent genetic changes in cells within a living patient's body, is subject to a regulatory framework that is still being developed and refined. The FDA's Office of Tissues and Advanced Therapies, which oversees cell and gene therapy products, has been engaged in ongoing dialogue with the CRISPR therapeutic community about the standards that will be required for in vivo editing agents targeting solid tumors. The core regulatory concerns center on three areas. First, off-target editing, the possibility that the CRISPR machinery will edit unintended genomic sequences in addition to the intended target, with consequences ranging from inconsequential to carcinogenic. Second, germline editing risk, the theoretical possibility in an in vivo setting that editing machinery could reach reproductive cells and make heritable genetic changes, a category of genetic modification that is subject to its own regulatory and ethical framework. Third, long-term safety monitoring, the question of how patients who receive in vivo CRISPR edits should be monitored over time for delayed adverse effects that might not become apparent for years or decades. The germline editing concern, in the context of a solid tumor therapy delivered to a cancer patient typically in later life, is considered by most regulatory scientists to be a theoretical rather than practically significant risk, particularly with delivery systems designed for tumor-specific targets. But it is a concern that regulatory agencies take seriously as a matter of principle, and the preclinical data required to address it adds substantially to the development timeline. The economic question is perhaps the most uncomfortable to discuss, but it would be intellectually dishonest to ignore it. CRISPR-based therapeutics are, by their nature, expensive to develop and potentially expensive to administer. The manufacturing complexity of biological delivery systems, extracellular vesicles, or engineered lipid nanoparticles, combined with the personalized or semi-personalized nature of some CRISPR therapeutic approaches, creates a cost structure that could make these therapies inaccessible to the majority of global lung cancer patients. Lung cancer is a global disease with its highest burden in low- and middle-income countries, where platinum-based chemotherapy is often the only available treatment, and comprehensive genomic profiling is unavailable. A CRISPR therapeutic accessible only to patients at major academic cancer centers in wealthy countries would, if successful, create a new category of molecular inequality in cancer medicine, effective for some, invisible to most. This is not a reason not to develop the therapy. It is a reason to think carefully, from the beginning of development rather than as an afterthought, about access, manufacturing scalability, and the policy frameworks that will determine who benefits from a successful outcome. Several researchers in the field, including members of the San Diego team, have stated publicly that global access considerations should inform licensing agreements, manufacturing partnerships, and pricing strategies for any CRISPR therapeutic that reaches the market. Whether those statements translate into binding commitments, and whether the commercial realities of pharmaceutical development allow for equitable access models, remains to be seen. There is one more hard question that deserves direct address, because it is the question that sits beneath all the scientific and ethical debates and gives them their urgency. It is the question of time. Cancer patients do not have the luxury of waiting for science to resolve all its uncertainties before they need treatment. The patient in Ohio who emailed Dr. Chen after reading the AACR presentation summary, the one whose question Chen described as the most affecting he received, was not asking whether the mouse data would replicate in humans. She was asking whether she would live long enough to benefit from what the mouse data suggested was possible. That tension between the time required to do science responsibly and the time available to patients who need answers now is not resolvable. It is a permanent feature of medical research, and acknowledging it honestly is more useful than pretending it away with either false urgency or excessive caution. What responsible science can do, what the researchers in this story have been trying to do, is move as quickly as rigor allows, be transparent about what is known and what is not, design trials that generate reliable answers efficiently, and apply existing knowledge to current patient care even before experimental therapies are available. The scientific debates described in this chapter are not delays. They are the process by which unreliable findings are distinguished from reliable ones, by which mouse data is tested against human biology, by which safety signals are detected before they become patient harms. The skeptics who press the hardest questions are not obstacles to progress. They are part of the mechanism by which science produces trustworthy answers. And trustworthy answers in cancer medicine are the only ones worth having. The horizon. There is a photograph that hangs in the hallway outside Dr. Michael Chen's laboratory at the University of California, San Diego. It is not a photograph of a scientific breakthrough or a celebrated experiment. It is a black and white image of a hospital corridor taken sometime in the 1950s, lined with iron lung machines, the negative pressure ventilators that kept polio patients alive by breathing for them when their respiratory muscles had been paralyzed by the virus. The photograph is not there as a monument to suffering. It is there as a reminder of distance traveled. Because the children in those iron lungs, the ones who survived, lived to see the Salk vaccine eliminate polio from most of the world within a decade of that photograph being taken. They lived to see the impossible become routine. Chen has never explained publicly why he chose that particular image for that particular wall. But anyone who has spent time thinking about the arc of medical history understands the implicit argument. The distance between a laboratory discovery and a transformed clinical reality is not fixed. It is not predetermined. It is shaped by the quality of the science, the urgency of the need, the wisdom of the regulatory and institutional decisions made along the way, and sometimes, not always, but sometimes, by a convergence of circumstances that accelerates what might otherwise take generations. Whether the NRF2 story represents one of those convergences is a question that the next several years will answer. What can be said with confidence today is that the scientific foundation is more solid than it was 5 years ago. The clinical urgency is well established, and the research community engaged with this problem is larger, more coordinated, and better equipped than at any previous point in the history of NRF2 biology. That is not a guarantee of success. It is a reason for serious, calibrated. Let us be specific about what the next several years are likely to look like, based on the realistic trajectories of clinical development as they stand in early 2026. The most immediate milestone, the one with the clearest timeline and the most direct implications for whether the CRISPR NRF2 approach will enter human testing, is the investigational new drug application that the San Diego and MD Anderson collaborative team has been preparing for submission to the FDA in the first half of 2026. An IND application is not a request for approval to treat patients. It is a request for permission to conduct clinical investigation, to open a Phase 1 trial in which a small number of carefully selected patients will receive the experimental therapy under closely monitored conditions, with the primary goal of establishing safety parameters rather than demonstrating efficacy. Phase 1 trials typically enroll between 15 and 45 patients, depending on the dose escalation design, and are designed to identify the maximum tolerated dose, characterize the safety profile, and gather preliminary pharmacokinetic and pharmacodynamic data. The FDA review process for an IND application typically takes 30 days, after which the agency can either allow the trial to proceed, place it on clinical hold pending additional information, or request specific additional preclinical data before the trial can open. For a novel modality like in vivo CRISPR editing in solid tumors, a category for which regulatory precedent is limited, the dialogue with the FDA prior to IND submission is typically extensive and iterative, and the San Diego team has been engaged in pre-IND meetings with the agency since mid-2025, according to statements made at the AACR annual meeting in April 2025. If the IND is filed in the first half of 2026 and receives a standard 30-day review without clinical hold, Phase 1 trial initiation could potentially occur in late 2026 or early 2027. The first cohort of patients, those receiving the lowest dose level in the dose escalation design, would likely be individuals with confirmed KEAP1 NRF2 mutant non-small cell lung cancer who had failed at least 2 prior lines of standard therapy. These would be patients for whom standard treatment options had been exhausted, consistent with the ethical framework of Phase 1 oncology trials that appropriately reserves first-in-human testing of experimental therapies for populations with limited alternatives. Safety data from the first dose cohort would require approximately 3 to 6 months of observation before the Trial Safety Monitoring Committee could recommend dose escalation to the next level. A complete Phase 1 dose escalation study, assuming no serious unexpected safety signals requiring trial modification, might generate primary safety and dose-finding data within 2 to 3 years of trial initiation, meaning, on an optimistic but not unrealistic timeline, preliminary human safety data could be available by 2029 or 2030. That timeline feels long when stated in years. It feels different when you consider that the first cell culture experiments demonstrating NRF2 deletion sensitivity in resistant lung cancer cells were conducted in 2021, meaning the journey from initial laboratory observation to first in-human data, if this timeline holds, would cover approximately 8 to 9 years. In the history of cancer drug development, 8 to 9 years from laboratory discovery to human safety data is not slow. For a novel genetic medicine targeting a challenging solid tumor via a delivery system that did not exist in its current form 5 years ago, it represents genuine scientific velocity. Parallel to the CRISPR Phase 1 timeline, several other developments in the NRF2 clinical landscape are expected to mature over the same period. The MD Anderson trial evaluating trametinib plus docetaxel in KEAP1 co-mutant KRAS mutant lung cancer, which is actively enrolling as of early 2026, is expected to report primary efficacy data from its Phase 2 expansion cohort in 2027 or 2028, depending on enrollment pace and event rates. This trial does not use CRISPR. It uses approved drugs in a molecularly selected population. If it shows meaningful activity in the KEAP1 mutant subgroup, it would represent the first prospectively validated targeted treatment strategy for this population using currently available agents, an important near-term clinical advance that does not require waiting for gene editing technology to mature. The ETO-PLUNG European Consortium trial, currently in protocol development, is expected to open enrollment in 2027, pending regulatory approvals in participating European countries. Its design, comparing standard pembrolizumab-based therapy to an NRF2 pathway-targeted experimental arm, will provide the first randomized clinical trial data on whether molecularly stratified treatment selection in KEAP1 NRF2 mutant lung cancer improves outcomes compared to current standard approaches. The cystinase program at Aterion Therapeutics, targeting the cysteine dependency of NRF2 high tumors, is also expected to advance toward clinical testing in this period, with a potential IND filing timeline of late 2026, according to the company's publicly stated development plan. By the early 1930s, meaning within a decade from the original San Diego cell culture experiments, the clinical picture for KEAP1-NRF2 mutant non-small cell lung cancer is likely to look substantially different from today. Multiple trial readouts will have been generated. The CRISPR approach will have either demonstrated preliminary human safety and early efficacy signals, or encountered obstacles that redirect the field toward alternative strategies. The pharmacological and metabolic approaches will have matured through their own clinical trial trajectories, and the molecular stratification of lung cancer patients by NRF2 pathway status will almost certainly be standard clinical practice, rather than the selective, center-dependent phenomenon it is today. This is not a prediction that any of the specific therapies currently in development will succeed. It is an observation about the direction of travel, that the field is moving toward a clinically actionable understanding of NRF2-driven resistance, and that the pace of movement is accelerating rather than slowing. But the horizon of this story extends beyond non-small cell lung cancer, and the broader implications deserve careful examination, because they represent both the greatest potential and the greatest uncertainty in the NRF2 narrative. The multi-cancer expansion experiments underway in the San Diego and MD Anderson Collaborative Program, testing CRISPR NRF2 deletion in preclinical models of hepatocellular carcinoma, head and neck squamous cell carcinoma, bladder cancer, and esophageal cancer, will generate data that either supports or challenges the pan-cancer therapeutic hypothesis that the Broad Institute genomic analysis raised. If the chemotherapy resensitization and immune microenvironment remodeling effects observed in lung cancer models replicate consistently across these other tumor types, the implications are substantial. It would suggest that a CRISPR NRF2 deletion strategy, or any therapeutic approach that effectively suppresses constitutive NRF2 activity, could address drug resistance and immune evasion simultaneously across a significant fraction of all solid tumors. The global disease burden potentially addressable by such an approach, measured in patients rather than statistics, would run into the hundreds of thousands annually. If the effects are lung cancer specific or limited to specific genomic contexts, as the German Cancer Research Center group has cautioned, then the therapeutic target remains important, but the scope is more circumscribed. The strategy works in lung cancer. It may work in some other cancers. It does not generalize to all solid tumors with NRF2 pathway mutations. The honest answer is that we do not yet know which of these scenarios is correct. The preclinical data from other tumor types will provide an initial indication. The clinical trials that follow, years from now, will provide the definitive answer. What is already certain, regardless of how the cross-tumor translation plays out, is that the mechanistic insight at the core of this story, that cancer uses a single master regulatory gene to simultaneously defend against chemotherapy, suppress its own death programs, and disable the immune response around it, is a genuine contribution to the scientific understanding of malignancy. That insight has value independent of whether the specific CRISPR therapeutic approach succeeds. It changes how we think about resistance. It changes what questions we ask about why some tumors respond to immunotherapy and others do not. It changes the vocabulary of the conversation between molecular oncology and clinical medicine. Science does not always succeed in producing the therapy it is aiming for. It almost always succeeds in producing understanding in medicine is the raw material from which therapies eventually emerge. Even if the path from one to the other is longer and more indirect than anyone would choose. There is a dimension of this story that has not yet been addressed directly, because it tends to make scientists uncomfortable and journalists overconfident. It is the philosophical dimension. The question of what it means at a deeper level that the human genome contains within it both the mechanism by which cancer kills and the potential tool by which cancer might be defeated. CRISPR is, at its origin, a bacterial immune system. Bacteria evolved it over billions of years as a way of recognizing and cutting the DNA of invading viruses, storing fragments of viral genetic sequences in their own genomes as a kind of molecular memory, ready to deploy precision DNA cutting machinery against any virus that matched the stored sequence. Humans, or more precisely, the researchers who recognized what bacteria had spent billions of years perfecting, took this mechanism and turned it into a tool for editing the human genome with a precision that was, 20 years ago, the province of science fiction. The gene being targeted, NRF2, is itself a product of billions of years of evolution, refined to protect cells from oxidative damage in an oxygen-rich world. Cancer, that most ancient of cellular diseases, found a way to hijack NRF2 and turn it from a cellular guardian into a cancer survival engine. And now researchers are proposing to use a bacterial immune mechanism, repurposed as a molecular scalpel, to disable the evolutionary hijacking that cancer performed on a gene that evolution built to protect life. There is something vertiginous about that recursion. Evolution fighting evolution, with human ingenuity as the intermediary. And it is worth sitting with that vertigo for a moment, not because it changes any of the science, but because it illuminates something important about the nature of the enterprise. Medicine is, in the deepest sense, the project of using what we understand about biology to intervene in biology, to redirect natural processes that cause harm toward outcomes that preserve life. CRISPR cancer therapy is that project operating at its most intimate level, inside individual cells, at the level of the genetic instructions that determine what those cells do and whether they live or die. The implications of that intimacy extend beyond oncology. The tools being refined in the context of NRF2 deletion for lung cancer, the delivery systems, the guide RNA design methods, the off-target editing assessment technologies, the clinical trial frameworks for in vivo gene editing, are tools that will be applicable to other genetic diseases, other therapeutic targets, other clinical contexts that we may not yet have imagined. The Phase 1 trial, when it opens, will be a cancer trial, but it will also be one of the first systematic human explorations of in vivo CRISPR editing in a solid tumor, a scientific event whose implications will extend far beyond the specific patients enrolled and the specific gene being targeted. What does all of this mean for the person watching this video? The person who may have a family member with lung cancer, or who works in a cancer clinic, or who simply finds themselves in the audience of this remarkable moment in the history of medicine. It means several things, stated as plainly as possible. It means that KEAP1 and NRF2 mutation testing in non-small cell lung cancer is a question worth asking today at any major cancer center as part of standard comprehensive genomic profiling. The testing exists. The clinical significance of the results is real, if still being defined. The information has immediate relevance to treatment discussions. It means that clinical trial participation for patients with KEAP1-NRF2 mutant lung cancer who have progressed on standard therapy is likely the most scientifically rational treatment option available. Not because experimental therapies are certain to work, but because the evidence for standard options in this specific population is weak enough that the uncertainty of a trial is a reasonable trade-off for the possibility of benefit. It means that the CRISPR NRF2 approach, whatever its ultimate clinical fate, represents a genuine scientific achievement, a mechanistically coherent, experimentally rigorous, independently supported framework for understanding a major source of cancer drug resistance, with a therapeutic strategy rationally derived from that understanding and in early-stage clinical development. And it means that the story is not finished. The most important experiments have not yet been done. The patients who will determine whether the mouse data translates to human benefit have not yet been enrolled. The Phase 3 trials that will ultimately determine whether this approach changes standard of care are years away, contingent on Phase 1 and Phase 2 results that do not yet exist. This is science in progress, not science completed. The distinction matters enormously. The iron lung photograph in Chin's hallway is, among other things, a study in humility. The children in those machines were kept alive by a technology that, within a decade, was rendered obsolete by a better one. The researchers who built those iron lungs were not failures. They were the bridge between a world where polio paralyzed and killed and a world where it did not. Their work mattered even though it was superseded. The CRISPR NRF2 approach may itself be a bridge, a first-generation strategy that opens the clinical pathway, generates the human safety and efficacy data, and demonstrates the principle, even if the specific technology is later refined or replaced by something more effective or more accessible. In medicine, first-generation therapies that prove a concept are not consolation prizes. They are how second-generation therapies become possible. Or it may be more than a bridge. The data, honestly assessed, does not yet tell us which. The human trials will. What is not in question is the importance of the question being asked. 1 in 5 cancer deaths worldwide is a lung cancer death. The fraction of those deaths attributable to drug resistance, to the evolved capacity of tumors to survive the treatments directed at them, is enormous. The fraction of that resistance driven, at least in part, by NRF2 pathway dysregulation may be in the range of 15 to 30% of non-small cell lung cancers alone. If a therapy directed at that mechanism can restore drug sensitivity, reactivate immune responses, and improve outcomes for even a fraction of those patients, the human significance of that achievement measured not in publications or patents, but in lives, would be profound. The horizon is not a guarantee. It is a direction. And the direction, right now, is forward. One last thing before this investigation closes. Science is not a spectator sport, and the questions raised by this research are not exclusively for scientists to answer. Should in vivo CRISPR editing of somatic cells be regulated differently from ex vivo editing? How should health systems prioritize access to expensive molecular therapies in a world where the patients with the greatest burden of lung cancer often live in countries with the least capacity to pay for them? What obligations do publicly funded research institutions have to ensure that discoveries made with taxpayer money reach the patients who need them, regardless of geography or income? These questions do not have molecular answers. They have political, ethical, and institutional answers, and those answers will be shaped by the choices of policymakers, regulators, insurance systems, pharmaceutical companies, and civil society organizations over the coming decade. The science can produce a therapy. Whether that therapy reaches the patients who need it, all of them, not just the well-insured and the geographically fortunate, is a question that begins where the laboratory ends. That is the real horizon, not just what we can discover, but what we choose to do with what we discover. And that question, the question of what we owe to each other in the face of knowledge we have worked hard to earn, is, in the end, the most important one this story raises. The comment section is below. The conversation is worth having. And the geographically fortunate is a question that begins where the laboratory ends. That is the real horizon, not just what we can discover, but what we choose to do with what we discover. And that question, the question of what we owe to each other in the face of knowledge we have worked hard to earn, is, in the end, the most important one this story raises. The comment section is below. The conversation is worth having.